to faster microprocessors that consume less energy and batteries that last 10 times longer, we can see that nanotechnology is arguably a discipline with all prerequisites to take us to next industrial revolution. Let us unveil the various potentials of these nanoparticles by commencing this program with a heartwarming musical note. I request everyone to rise for Tamil Thayavalthu.
நீராறும் கடலொடுத்த நிலமடந்த கெழிலொழுகும் சீராரும் வதனமென திகழ்வரத கண்டமிட்டு தெக்கணமும் மது சிறந்த திராவிடனல் திருநாடும் தக்க சிறு பிறை நுதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிலமை திறம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
Thank you, and you may be seated now. I'm extremely privileged to invite distinguished personalities. I request all the dignitaries to kindly come on stage and accept our floral welcome. Our patron professor is Meenakshi Sundaram, Center for Biotechnology, Anna University. Our chief guest, Padma Bhushan Professor T. Ramasamy, former Secretary, DSC, Government of India, former Director, CLRI. Our Chief Advisor, Dr. C.N. Ramchan, co-founder, CEO, and CSO, Maggenome and Theragen Group of Companies. Our guests of honor, Professor R.V. Upadhyay, Provost, Charotar University of Science and Technology, and Sri Abhay Kumar Sri Srimal, founder and chairman, NanoLife. I also invite our convener, Dr. Priyanka Samji, Theragen Biologics Private Limited, and our organizing secretary, Dr. Kamalika Roy Chowdhury, Maggenome Technologies Private Limited, onto the stage. I also invite our patron, Mr. Sam Santosh, chief guest, Dr. T.P. Singh, and Dr. Ajay Kapoor, who have joined this inauguration through online. I request all the guests to kindly accept our floral welcome. Thank you all. Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world. May I now request our renowned chief guest, Padma Bhushan Professor T. Ramasamy, to illuminate the event by lighting the lamp. I request all the guests of honor and dignitaries on the dais to accompany him. Thank you all. May I now invite Professor S. Meenakshi Sundaram, Center for Biotechnology, Anna University, to deliver the welcome note.
Good morning to all. Respected Padma Bhushan Sri Dr. T. Ramasamy, Dr. R.V. Upadhyaya, Sri Abhay Kumar Srismal, and also by online Dr. T.P. Singh, Professor Ajay Kapoor, and Sri Sam Santosh, Dr. Ramchand, speakers of this conference and delegates. I am happy to welcome you all for this one-day conference on biological applications on nanoparticles. We feel extremely honored by the presence of Padma Bhushan Dr. T. Ramasamy, former Secretary DST, who is one of the successful scientists in translation research on this occasion as chief guest. On behalf of the organizing committee and Anna University, I welcome you, sir. It's a pleasure for all of us that distinguished professors Dr. R.V. Upadhyaya, Provost, Charter University of Science and Technology, Dr. T.P. Singh, Sir Distinguished Fellow, and Professor Ajay Kapoor, Vice Chancellor, Chinmaya Vishwa Vidyapith, to be with us today in this conference. We welcome you, Professor. I'm happy that Sri Abhay Kumar and Sri Sam Santosh, the successful industrialists in translation research, to be with us today to share their thoughts. I extend warm welcome to both of you. I also take this occasion to extend a warm welcome to all the distinguished speakers, delegates of the conference, and Dr. C.N. Ramachandran and C.N. Ramchand and his team from MagGenome. Welcome you all. In initial stages, nanoparticles were studied because of the size-dependent physical and chemical properties. Now, they have entered a commercial exploration period, and it has variety of applications in biology and medicine, from fluorescent biological labels, drug and gene delivery, separation and purification of biological molecules and cells, MRI contrast enhancement, etc. Today, in this one-day conference, we will be listening to some of the applications of nanoparticles in biology. Translation research is to move basic science discoveries more quickly and efficiently into practice. Most of our academic research are ending with high quality publications or process development to 80% of the technology readiness levels of commercialization. Realizing this, DST has initiated a program, Technology Enabling Centers, at various universities across the country. Anna University is one among such center supported by Department of Science and Technology, Government of India. The objective of these technology enabling centers is to improve the TRL levels of the academic research to commercialization stage and successful commercialization. I'm happy that MagGenome was efficiently and successfully doing that, and really we would like to learn from them the art of translational research. Once again, I welcome you all for this one-day international conference on biological applications on nanoparticles. Thank you.
Thank you, sir. May I now, I would like to call upon stage Dr. Priyanka Samji, Senior Research Scientist, Tarajan Biologics Private Limited, to present theme of the conference.
Good morning, everyone. On behalf of the organizing committee, it's a great pleasure and honor to invite you all to join the ICANBio 2023 conference, which is based on the biological applications of nanotechnology. This is indeed a great opportunity for all of us to be a part of these scientific sessions and listen to the leading experts about the advantages, the applications and innovations in the field of nanotechnology. In 1990s, when we used to talk about nanotechnology, people used to make comments that nanotechnology is just a hype. As the name suggests, it's nano, so the future is also nano. However, now we all know that nanotechnology is a proven technology. And if we look at the projections for this particular field, in 2020, it is actually around 1.76 billion US dollars. And in 2020, 2030, it is predicted to be $33.63 billion. So you all can imagine from where the nanotechnology field has gone to such a huge level. Now, the overall idea of this conference is to advance the field of nanotechnology by fostering the interdisciplinary collaboration and promoting the exchange of knowledge and ideas. It's actually a networking event which brings together the inspired nanotechnology researchers and scientists to listen, discuss, and share their knowledge. Now, integration of nanotechnology into the biology has revolutionized the development of biomedical devices, the implants, the diagnosis, the live imaging, the targeted drug delivery, the regenerative medicine, tissue engineering for the treatment of diseases and injury. Now, this conference will be a multidisciplinary forum for discussions and deliberations on bridging the gap between the fundamental research and the application in engineering, biology, and medicine. In today's event, we will see the convergence of different fields of science. We will bring together the physicist, the chemist, the biologist to discuss the latest developments in the processing of nanomaterials and the challenging in the applications of these in the field of engineering and biomedical field. It will be a great opportunity for all of us for interaction and collaboration between the academia and the industry to extend the lab or pilot scale research in the field of nanotechnology to develop into innovative and cutting edge technology for real deployment and solutions. So in this conference, we'll have different sessions which will cover different aspects on the synthesis, characterization of different types of nanomaterials, the trends, the advances in the field of nanotechnology, and their applications in engineering, diagnostics, treatment of tumors, biomedicine, and biotechnology. We will also have the launches of various MacGenome products during this conference. So with this, I would like to thank all of you to be a part of this one-day event, and I'm sure we all should take this best opportunity to shape and design our future in the field of nanotechnology. Thank you.
Thank you for the introduction, ma'am. May I now request Dr. C.N. Ramchan, co-founder, CEO, and CSO, MacGenome and Teragen Group of Companies, to address the gathering.
Good morning, and thank you very much. Thank you, Professor T. Ramaswamy, whom I met. I met him several months back, but then just invited him 2 days back, and he has kindly consented to be the chief guest. So he is Dr. Abhay Kumar ji, and I talked to him about 2-3 days back, and immediately he came from Pune and agreed to be here. Thank you very much, because he also has a company in nano sciences called NanoLife. In fact, I sent earlier him multiple messages and the invitation, but he did not get. Only 2 days back. Anyway, thank you very much. And I'm extremely thankful to Professor R.V. Upadhyaya. He'll have to talk a lot, but I don't have time. So he is Professor Meenakshi Sundaram, and also our organizing people, and from online, Professor T.P. Singh, Professor Ajay Kapoor, and especially Mr. Sam Santosh, our chairman, who is the driving force behind all the things. Thank you, everybody, for joining. So I'm not going to talk much about any of the technology which you are going to hear, but I want to tell that some genesis of this MacGenome technology is a very good example of a translational research which happened in the university. This was not my field. I was not either working on nanotechnology or similar things or product development. I was a university researcher teacher, both in US and UK and in India. So back in 1980s, middle of late 80s, when I was working in University of Wales, so I was working on totally different mechanisms of schizophrenia, depression, mania, and product development related to that. During that time, Professor R.V. Upadhyay, he was a professor at that time, Bhavnagar University, came over to University of Wales, and it so happened that I was the patron for the students over there, because there are not many students, hardly any teachers in University of Wales. And being a physics people, and my background is chemistry, physics, mathematics, I was so interested talking many things. Then they talked about this nanoparticle, magnetic nanoparticles, and their main work is connected to the applications in physics, engineering, and related things. So during that time, some of the characteristic they told that, how the nanoparticle, various sizes, hydrophobic, hydrophilic site, dielectric constants. Then I also suggested that a lot of things can be done in biology. We initiated, because my laboratory was very near to the physics lab over there. Then we used to collaborate. Even Professor R.V. Mehta, I remember that both of them used to come to my lab, and we used to work together. Can you imagine? In 2 weeks' time, we completed the work, and in the next 1 month, we published that paper. And one of the landmark paper, which is even now quoted very high, how proteins or other molecules can be linked to nanoparticles. This was the basic research, and subsequently, it's a story. Then what has happened, because of them, I got collaborated with various groups in Slovakia, Poland, Czech Republic, and many other places. And a lot of PhD students came up. Then subsequently, I came to India. I started a couple of PhD students when I was in Sun Pharmaceuticals. So I was heading the research for Sun Pharma at that time. And I was allowed to work on nanotechnology, totally different academic research. And one of the first students in nanotechnology was Sayed. And unfortunately, he's in San Francisco. He could not join. And, the very base work of this particular technology was initiated, and he published over the period of his PhD about 12 papers, multiple patents. So that was the story behind. And subsequently, we started various, in the due course, until the company was started in 2014, many things we have done. Then, 2007, I was invited by our Prime Minister, that at that time he was the Chief Minister, and the start of the Vibrant Gujarat. I was one of the invitees to talk on nanotechnology, and it was about 2,000 people, 2,000 corporate people he wanted to address, and that was the start of it, and various opportunities. I remember telling, subsequently 2 or 3 years I went there to talk also in Vibrant Gujarat, and they started a nanotechnology program in Gujarat. Even now the funding is going on, isn't it? Gujarat Biotechnology Mission. Anyway, this was the start of the things. Then, but then, anybody can, one of the thing I realized is that discovery is not the great thing. Even development, I always tell, anything can be discovered and developed, especially in drug discovery, biologics discovery, chemical discovery, any of these things. And development also is not great thing. Two major problems we are going to face is, one is the regulatory problem. And even if you are passing through the regulatory, how to sell it? That is the greatest problem what we are facing, because it's not easy thing. Especially if you are using a different technology, a better technology, somebody is already using, that is the challenge you are going to do. And let me tell you, if anyone of you are involved in startup community, this is the first thing you will have to see, how to sell the product. Now, 2014, we have started, and Sam was very optimistic about when I talked to him, and he invested, he encouraged. Then we started the nanotechnology company called MagGenome, started in Cochin. First 3 years, we various products we developed. We could not sell a single thing, let me tell you. But subsequently, and perseverance, lot of try. But now, about 40, 45 products we sold. And the most important thing is that during COVID period, all our sales, whatever little we are selling, everything came down, because nobody, no research, nothing was there. First, we started the research product. Then we introduced COVID, that viral transfer medium, molecular transfer medium, RNA extraction, and RT-PCR. The major problem was not developing the kit, but getting the approval. 6 months we ran to get the approval. Finally, we got probably one of the best kits ever. You can see that, probably Andrew will be showing that one and how good the product is. Then we launched it, and in 9 months, we could sell almost in India 6% of the COVID kits. And because of us, the cost of whether it is RNA extraction or the entire RT-PCR, the whole kit came down from 2,000-3,000 to 300-400 rupees, because the technology what we have used, the unique technology using nanoparticle, and that was probably the best in terms of quality, quantity, then no shearing, and I will tell you, 25 million kits has been sold, not a single recall is there. So, and the same way we have now introduced various other products, so many different types of products. One of the greatest product, I think what we are going to make a big is the size selection. Not many companies have got a unique technology, and using our technology, we have developed. So let me also tell you one thing that, in the last few years, we have several patents, multiple publications, 30, 35 publications, and we got DSIR approval, multiple awards. We could bring one of the Nobel laureates to our laboratory for two or three times. Major conferences we organized, and this is the continuation of the last conference, Icon Bio, 1917. Now this is, 2019. And now here, what you can see is that you can various types of scientific talks, but we have given only 10 to 15 minutes, everybody. We also have various product launches. Now, the most important thing is that, all these nanotechnology or the products, the major place is US. So we have set up an office there. We have few people working there, and we hope that we will be able to sell in a big way in the US. That is our aim. And eventually, we think that all over the world, we will be able to introduce. We have also introduced certain medical devices, saliva collection kit, fecal matter collection kit, and RNA extraction or DNA extraction, high throughput. All those things we have introduced, or you will be able to hear in between, and we will be launching this product. In the meantime, you will be able to hear a lot of very good talks. And thanks to all the organizers, the first time I have suggested some of the junior people, because if you remember AMDP conference, which was happened 2 or 3 months back, we had a tremendous response, 930 people, and it was always full. But this time what we have done is that, this is 90% of the people are online. Only limited people we have the access to offline. There are various reasons, because of everything we started about few days back only. So thanks to Kamalika, then Arunabha, then Priyanka, Anirudh, and Sri Priya, all the team for helping us, and especially Professor R.V. Upadhyay for, introducing this technology to me, physics technology. Thank you very much, and you are going to hear a lot of good talks today. Thank you. Thanks a lot.
Thank you, sir. May I request Mr. Sam Santosh, Patent Chairman, Cygenome and Maxgenome and Managing Trustee, STRF, who is joining us online to share a few words.
Hello, can you hear me? Can you hear me? Good morning, everybody. Respected and distinguished guests on the dais, all attendees at the conference who have been able to come in person, and I can see over the 150 attendees offline. Thank you all for attending this conference. I would have also liked to be in person, but unfortunately, I'm here in San Francisco, so I'm forced to attend, online. I hope I'm audible.
Yes, we can hear.
Thank you, Dr. Ramchandran. So you all heard the from the earlier speakers the goal of the conference and the main objectives that we hope to achieve by bringing all of you together, and the wonderful speakers that you will hear in the rest of the day. And this is a very important event, I feel, in the sense that biological application of nanoparticles, especially uncoated iron nanoparticles, has not been well accepted or well known across the world. And the pioneers in this are Dr. Bhatia sitting there, and Dr. Ramchand, and they have been able to bring this out and get successful products out. So it is not just like being uncoated is such a unique thing on its own, but the fact that you have to keep in mind is that that allows many other benefits, such as lower cost, better action, and also better value to the environment in reducing the toxicity and waste that occur from most of the chemical products. So in this way, Maggenome, through Ramchand's patents and the work of the scientists, have been able to develop a set of products. And Dr. Meenakshi Sundaram was earlier kind enough to note that Maggenome and our companies have been successful in translating research, to products. But I would like to point out that translating is not easy. So it not only it needs perseverance, but it also needs a lot of time. As Ramchand briefly pointed out, 2014 the company we started incubating, but it took up to 2018, nearly 4 years before we could really get products which would be accepted by the market. So it takes a long time. The bar is very high, especially in products in life sciences. So to reach that, we will need we meaning the whole industry will need the support of the government and the financial institutions, because especially the financial institutions like the venture capitalists or other funding agencies typically are often looking for focusing on much short-term gains. So in these type of companies, that is not practical. So there has to be a separate approach and a separate funding mechanism to take into account the needs of companies in the life sciences industry. So unless that is there done, India will still always struggle in converting research in life sciences into successful products. So this becomes even more important as we go forward. Why? Because as you all know, this is the century of biology. So a lot of the problems that has been created by the industrial revolution, which also brought in a lot of benefits, but at the same time, the it has created pollution. It has created so much waste. If you start from plastics, to all the other fertilizers and the drain offs to the oceans, and so all the pollution and all the damage that has caused to the earth and to our world and to our atmosphere, including global warming, all has a potential to be solved using through biology. So life sciences industry is going to be the most critical industry in this century, in the current century. And India, as a growing economy, which is currently at the fifth place and will reach the third place maybe in the next 10-15 years, uh, at the same time has a large population. So unless we change our approach from the polluting technologies of the past to environmental friendly bio technologies of the future, there is no way that Earth can survive. So this is not just a hype. I mean, it is reality. But at the same time, the great progresses in genomics through genome sequencing that we saw in the last 15 years, the huge progress that we will see now with the proteomics, a lot of revolutionary developments are happening in proteomics, which will all, both this genomics and proteomics, will lead to a significant growth of synthetic biology and biomanufacturing. So that is the wave of the future, which I typically call the bio wave. So Maggenome has done its part, a little part, in pushing this wave, and we continue to I mean, we hope that we can continue to develop, more products, as Dr. Ramchand mentioned, in genomic sequencing. Site selection becomes a very important step, before you start preparing the library. So site selection currently, there are only a couple of products in the world which are very expensive. So Maggenome has introduced a product which is equal or better, and which is maybe half the price. So not only reduction in price, improvement of quality, and make in India. So these are the goals of Maggenome. So I'm very happy that all of you are here, and I wish you all the best in hearing the next set of talks, and also interacting with all the speakers and amongst yourself. Because collaboration is going to be the key, and so collaborating in across different industries and different technologies will hopefully make India a better research center. Thank you very much.
Thank you, sir.
That such a process has begun as a country. But of course, I learned science not from the school. I learned science from nature. So while we celebrate these biological applications of nano, I like to remind all of us the time proven biological application of nanoscience is the way we sweat. Our perspiration itself is a consequence of very fine principles of nanoscience. We say, what is this man doing? The structure of collagen was solved right in this campus by J.N. Ramachandran. the triple helix and this and that. But the way skin is organized, 5 collagen molecules organized at the corners of a regular pentagon, and the cavity size is 1 nanometer. That's how skin is layered. When it is dry, when it is wet, it is 1 nanometer. When it is dry, it is 0.7 nanometer. So, a water molecule has to go through this pore, it cannot be liquid. It cannot be liquid at body temperature. Why? Because 2 water molecules have a hydrogen bond that occupy a space and 6.4 nanometers too small. So they have to therefore not, they have to forego the hydrogen bond structure which converted the gaseous molecule at liquid temperature, at room temperature into liquid because of the hydrogen bonding. This was known to nature before. That's why all of us became what we are. That's a true biological application of nanoscience. Unfortunately, we don't teach our phase rule in physical chemistry with this understanding. That's why we have come to understand these things in a different way. So a man discovers the nano features and then says, look, this is great, which is very good, very understandable. But looking at it from the perspective of somebody whose first principles in science, how they happen, when you have a structural confinement of a nanoscience, the properties will change. The gold will not remain yellow if the number of atoms is less than 300. So the color of the gold will change depending upon the number of particles assembled in the system. Therefore, there is a structural confinement which changes the properties, which changes the surface energies, surface area, and therefore a lot of functional properties. A nanoscience really is an aspect of science that elicits the value of the nano-organized particles in the structural processes for doing several things. Several things. Of course, changes in societies happen. There are episodic changes, there are periodic changes, there are gradual changes. In science also, all 3 things have happened. Gradual changes happen, episodic changes have happened, and periodic changes happen. But there are epochal challenges. A nano is going through this epochal phase of change in the post, what we call 21st century regime. Therefore, it is an aspect of science change that has opened up new avenues, new opportunities, which are hitherto unknown. And in the area of biological application, because nano has applications in several areas. I won't say only to biological. You can use it for waste degradation. Nanoparticles can happen. And after all, the so-called Tanga Paspam, Swarna Paspam or something they used to call, that was part of the nano applications of gold. But at that time, the underpinning science was not known. It was empiricism. And today, we have good tools to recognize certain aspects of that science and use it very well. And of course, as was pointed out by Dr. Ramchan, when you start applying these to biological processes, there is this, they call it in the kinetic processes, Demon. And this Demon is called regulation science. And this regulator is supposed to bring a certain control on this. It turned out that while I was still in the government, we tried to bring a regulatory policy framework, not regulatory laws. Because laws must follow regulatory policy in framework. Because the policies we learn. You implement something and something goes wrong in that policy implementation, then we know what we should not do. When you bring about law, you first should know what should not be done. That's why we have tried to bring the regulatory policy in the nano itself at that time. And it is one good thing about this. All of the whole world is in the same boat at the moment. as far as nanoscience is concerned. Everybody has the same problem. Having said all these very broader aspect of nanoscience, how does it appeal to a biologist who has a certain interest in the process? If you go and talk about a chemical engineering, they'll talk about self-cleaning application of nanoparticles. They'll say construction material you can use and you don't want to clean the floor, clean these walls. But for a biologist, we talk about controlled drug delivery. We talk about site-selected delivery, targeted delivery. And there is a certain level of controls which are not available in solution phase, which nanoparticles can provide. The solution can go everywhere, but nanoparticles can be targeted, can be delivered on a specific processes. And then talk about nanorobots. You can go to look at cancerous cells and break open this. There are several opportunities have opened up. Opportunities may be several, but converting that opportunity also will find a lot of challenges associated with it. Therefore, when Dr. Ramchand was narrating the challenges he had crossed, but Vivekananda said there's no success in life without challenges. Therefore, there has to be challenge in this process. Now, I would say he also talked about marketing. In fact, he said selling. Selling nano products is a problem. I would like to differentiate between selling and marketing. Selling is all about meeting an established demand. Marketing is about creating a demand for a product which is not known before. Therefore, you can't sell something which is not known before. You have to really create a value chain for it. Therefore, I think we removed the jargon of selling in this nano dimension and move to marketing. Because you have to create a demand for it. Then you have to create a demand. These ask a question, how do we get new value for it? So you need to look for, you should be able to deliver values that cannot be delivered today by known products and previously known systems. Delivering value is an important part. And that's where the science of nano comes into play. That is where you have to go back and say, Arey Baba, existing products cannot do this. And therefore, you are going to create something that other products cannot do. That is what I think the biological applications of nano should actually deliver. Having said this, my sister-in-law tells me her husband is a good teacher and I'm a good examiner. Therefore, I would like to play the role of an examiner and put a challenge in front of these nano guys looking for applications. Today, you take these autoimmune diseases. Okay? Autoimmune diseases. Any of the autoimmune diseases a cure? The truth is no. Absolutely no. Arthritis is non-killer disease, autoimmune disease. If there is some sort of a rehabilitation, some help is supposed to be in Ayurveda. Okay. At least it's a non-killer disease. Take GBM. Okay. That has a terrible consequence. The global basement membrane, the kidney has this system, it's type 5, type 4 collagen. And toxicants come and destroy the membrane, and the kidney doesn't function. And if you are able to transplant, with kidney replacement, transplant, generally the lifetime after the surgery is about 4 to 5 years, life expectancy. Here is a disease caused by some toxicant generated inside the body. And what is the procedure for it? Plasmapheresis. Take the blood and do a affinity chromatography and remove this. This is the process. And the plasmapheresis has a high degree of, let me say, danger for life risk. can I really create a value chain for the nanoparticles that I designed for management of autoimmune diseases? There are several other autoimmune diseases. Today, no allopathic treatment has a clean cure for autoimmune diseases. I would like you to examine the possibility of using those gold particles and nanoparticles, because the gold has an advantage which other element doesn't have. Because I'm a transitional chemist, I understand the difficulties of dealing with metals. Gold, it may be a good jewelry, but it has one property that's not easily reacted upon by many others. Therefore, you can use it as a carrier without the danger of the biological changes that happen inside the body, and use it as a carrier. And can I use very targeted systems to handle some autoimmune diseases for which there is no cure and people are dying after 4 to 5 years? How can you transplant, kidney transplant, the so-called regenerative medicine itself needs to undergo a different change. You can't take a organ out of a human being and put to another human being. I think its time has to come. Ask the question, can I really create, culture the organ outside the body? 3D printing of organs. And the 3D printing of organs, if it is possible, then can I use these nanoparticles that I set up to address the autoimmune diseases? I think we will break a new path. I leave this as a challenge. Thank you very much.
Thank you for the informative speech, sir. We will try to keep up ways to address your challenge. Our guest of honor is Professor Ajay Kapoor, Vice Chancellor, Chinmay Vishwa Vidyapeeth, Kerala, who has joined us today online. Professor Ajay Kapoor was rated within the top 2% of researchers in the world in his subject area. He served as Pro Vice Chancellor at Swinburne University. He is a Fellow of Engineers Australia and the Cambridge Commonwealth Society. Without much delay, I kindly invite our guest of honor, Professor Ajay Kapoor, to address the audience.
Hello, can you hear me?
Yes, sir, we can hear you.
Okay. Hari Om to everybody from Kerala, and it's so wonderful to be here with Ramchand, you, and many more distinguished people. And so nice to hear the journey, Ramchand, that you explained. And I can vividly remember maybe 20 years back when Syed, after that initial work, came to Sheffield and Newcastle universities, where I was, and started to work on that, and some of the small grants for commercializing we got from, I think it was maybe Sheffield, where we got that grant and we worked on that. Very good to see that how that whole work is now developing in this area. And also goes to show what Sam was saying earlier, that it takes a fairly long time to commercialize these things. And so wonderful to hear Professor Ramaswamy also mentioning about the new possibility. After my work in the UK and Australia, last year I came to Kerala, and this is a university which is very focused on Indian knowledge systems. So the knowledge that has been traditionally with India over thousands of years, how can we modernize that? And that is a topic which I will also like to touch upon. Ramchand, you are very knowledgeable about the very modern ways of looking at technology, looking at these particles and making sure that they are useful for the humanity, for the community. As I noticed that Ayurveda has got vast experience of billions of people over thousands of years. We may not understand the science from the modern perspective, but the experience base is there, which says that many things work. To give you very simple examples, bhasmas are there. There are various ways of producing them. One can take a herb, burn it, and then those are nanoparticles, which are very small, tens of nanometers in size. One could actually grind these things, but that grinding into a paste using a stone and then rubbing it on that can also give you nanoparticles of different sizes. And Ayurveda actually says that you have to produce these particles to that particular root. I don't think many modern ways of using these Ayurvedic medicines are doing it correctly, but nonetheless, those are those ways. And then how do you use them? I see regularly chandan being applied on body, ash being applied on body, being applied on the forehead. So as in nanotechnology, nanomedicine, we know that directed delivery, taking that medicine to that particular part of the body where it is useful is very important. Is Ayurveda getting it right in saying that you apply chandan on your forehead, you apply chandan in your neck, apply the chandan on your wrist? Are those things capturing something which science can find out how is it being done? I know that there are many ways of taking the medicine. Orally, those medicines can be taken. Nasya is another thing to clean the nasal, and then they have some kind of a fume, and then the person inhales that. Is that also some way where it goes into, does the medicinal work on a certain organ? So those are the open questions which science must tackle. We have got a lot of wisdom, we have got a lot of knowledge. Scientifically, we need to analyze and see, is there something that can be utilized and that can be made available for a much larger population. So coming immediately after Professor Swami, who also mentioned about these things, I'll also like to say that, Ramchand, and looking forward to work from you, your group, and seeing that what else you all are doing for the community. So with that, I like to stop. Thank you and say Hari Om again.
Thank you for the illuminating speech, sir. I now welcome our distinguished guest of honor, Dr. T.P. Singh, SERB Distinguished Fellow, AIMS, New Delhi, who is joining us online today. Sir has played an active role in the development of drug design in the fields of antibacterial therapeutics, tuberculosis, inflammation, cancer, and gastropathy. He has published more than 415 research articles and contributed more than 565 structures to Protein Data Bank. May I now request our guest of honor, Professor T.P. Singh, to address the gathering. Just wait for 2 minutes. We'll be connecting Sir online. Sorry for the delay. Sir would be joining us for the time constraint. I feel extremely happy to welcome Dr. R.V. Upadhyay, Provost, Charotar Institute of Science and Technology. Dr. Upadhyay does research in condensed matter physics, experimental physics, and materials physics. Without delay, I now request our guest of honor, Professor R.V. Upadhyay, to share a few words.
First of all, very good morning to one and all present in the auditorium as well as online. Distinguished Chief Guests, Padma Shri Professor T. Ramaswamy, Professor T.P. Singh, Dr. S. Meenakshi Sundaram, Dr. C.N. Ramchand, Mr. Sam Santosh, Professor Ajay Kapoor, Professor Abhay Kumar, and all those other dignitaries of the dais. It is extremely, I'm honored today that I have got an opportunity to be with Professor Ramaswamy, whom I had a privilege to invite him in our university as a convocation address. So I came to know from that time. And earlier I used to know him because I had few DST projects sanctioned under when you were the chairman of the part. He has rightly says certain things which I will be narrating the later part of my speech. In the global, the scientific world, the nanotechnology and nanoscience is the buzzword for all the researchers right now. The reason behind this is in all other researches, the distance between the fundamental discovery and the translation research is always takes a long time. But in this field, it has gone like in a fast pace. And one of the field where the needful contributions were made in the applied basics and nanoscience is a translation research and biological science. In this regard, MacGenome has created a very high impact combined with the public as well as inspired many researchers to do research in nanoscience and nanotechnology. So I take this opportunity, Dr. Ramchand, Anirudh Bhatti, and all his team of MacGenome for doing the nice work during the last few years. Now see the things rightly told by Professor T. Ramaswamy that it's a demand-based research is always. And from last couple of, I think, 20 years, in India, we have concentrated on basic science of research. And from last 5 years, I think DST has focused more on doing the translation research funding rather than doing the only pure basic science research. He's rightly told that 1,000 crores rupees has been established for nanoscience and technology, I think in year 2004 or 5, I think that he started it. Yeah, 2006. Yeah. So that time it was given for the large amount of projects were given for different applications. And I'm happy that because of that initiative of DST, today people have come up with a lot of research and a lot of field of research, different things. As Dr. Ramchand rightly told that in 91, 92, I had been to Commonwealth Fellow and I went to the UK at the University College of North Wales, where I met first time Dr. Ramchand and Mrs. Ramchand both at the same lab. My professor, actually, I am being a physicist, but I was selected in chemistry. So my lab was a physical chemistry lab. Having no knowledge of chemistry because I'm a hardcore nuclear physicist who developed the radiation shielding material for Bhabha Atomic Research Center, and we have sold to them as a know-how. When I went there, there was no option for me because I had to learn the chemistry. I was thankful to my professor there, Professor Stuart Charles, who is considered the pioneer in nanoscience research and development of magnetic nanoparticle. I started working with him, and he taught me a lot of tricks and doing it for nanoscience and nanoparticle synthesis. So later we had an Indo-UK alias program under the University Grant Commission. So I was visiting a number of times. But at that time I met Professor Ramchand, and I remember very clearly the incidents that I am a person who doesn't like to be on table sitting and doing the lab work always. So I was chatting with his eldest daughter, Rakshana, and I was playing with her because she was 2.5 years old that time. And my professor R.V. Mehta and Ramchand were sitting on a dining table and doing the very serious discussion on how to bind the nanoparticle with the enzymes, different enzymes and different proteins. So during that talk, he has spoken some few words, which has triggered me. And then I told her, okay, let's go tomorrow to the lab and try it out. And that is how the success came and we could publish the papers in a few years. Within no time, we could publish the papers. And that was the starting point of Nano. That time, I never thought that he will bring this whole technology to this level. So I feel proud that at least I have contributed 1.1% of his growth, whatever he has done so far today. It is. Coming back to the same research which we have continued from last 36 years in our university, I do believe that being in a Gujarat, we always think about research to rupee. Because in Gujaratis are business minded entrepreneurs, so they don't fund otherwise if you don't convert into research to rupee. So that was the reason why we have started thinking in that line. It's a very hard time, I'll tell you that, because doing research and publishing paper is a one part, but as Professor Ramaswamy said, that marketing is a different thing. And he's rightly pointed out the demand, because I cannot clean off everybody saying that, okay, you don't use this. If I say tomorrow that don't use petrol, use this liquid for your vehicle, nobody will going to use it. Same thing happened with us. We have started developing with a transformer coolant, which is we have got a power transformer with 100 kVA, 200 kVA. The biggest issue we had regulatory issues. We got all the sanction except the color of the oil. They don't like black color of my oil. So I got all the certificate from the IRDA, certified that it works fantastically well for coolant as a transformer. But because of the color, they cannot grant me to use it. And now I cannot change the color because it's a natural color what we have, right? So I cannot change the color of my particle because they're opaque light. So there was no option for me. Then we started different thinking, different way. In European countries, there is no limitation on the color of the particle, color of the liquids. So you can sell them. That is how we started selling the liquid to the US and UK and Japan. And this is starting we are doing from last 15 years. And no doubt we have put a price value of $1,500 a liter processor I sell to the US and UK even today, this liquid. Finally, my research colleagues, he told that it's not good to do this way. Let us try something different. And I'm proud to announce that next month I'm going to Slovakia. We are opening a company there to establish the nanoparticle liquid research for putting into the transformer of their whole company part of it. So finally, if at the last 20 years of our hard work, we are successfully implementing now in Europe to put in high power transformer as a coolant. Now this synthesis was a demand based why I said, because generally we use the transformer oil and nobody is going to replace the transformer oil. Now the similar way, Professor, I do agree with what you have to autoimmune diseases because it prevails more in US. 80% of the US population is affected by autoimmune disease, while 18% is in India. So it is not so far that we will be reaching to the part of that. I do agree with that. It is a requirement of the demand. There are few institutes in US, they are working on exactly what you told is nanoparticle based research on autoimmune diseases. But the thing is the acceptance. We don't accept it very fast unless until it comes from the outside the country. If US, UK people tell that this is good, then we accept as a good, including the scientist. We don't accept our own scientists who are good at, but unless it has got a tag of America's coming from US or something, we respect them more. This is what the changes we required in youngsters. We have the capability to do research very good. And I'm very happy that DST has funded so many research projects to so many centers for excellence across the country to establish the research. And they have got well-equipped labs. Only required is the dedications and hard work. I feel ashamed sometimes seeing Professor Ramaswamy working at even a big part. He's sitting here and spending your whole time for the research and doing the research. It's very encouraging and he's a role model for all of us to see that because we retired once we retired from the job. That's not the case with him. The second part of his what he has told is about how we can target the market values. I'm a teacher as a professor, but involved with so many business industries because finally we have to sell our products. So in our university, we have established Section 8 company now to incubatees for the incubator to come and put up the part. And I'm very happy that the place where I'm working right now, Charotar University of Science and Technology, though it's a private university graded A+ by the NEC, our management system, whole management is so supportive that Anirudh knows because he has graduated from our university for his PhD degree. The day we announced the Section 8 company, the same day our chairman announced 25 lakhs as a token money for start up the Section 8 company for incubators. And today that amount has raised within 6 months, it has gone to the 1 CR value for incubators to start up their companies in our institutes. So I welcome those youngsters or whoever wants to have the incubator, most welcome to the university for doing the research in this kind of things. The last thing is that I would like to tell you that nanoparticle, particularly magnetic nanoparticles research, is a key issue even for the people who synthesize this. And Professor Ramaswamy says rightly that to synthesize a coated particle having a monodisperse size is much easier, but to synthesize a nanoparticle without coating and having no agglomerations is an art. Because this is the difficulty everybody faces in all our cells. And then to control the size, right from 5 nanometer to 25 nanometer size range without agglomerating the particle. We can disperse them in a hydrophobic or hydrophilic carriers, even without using a surfactants. We can give a charged particle, and whenever you want to extract, you can use it. So I'm proudly say that the department at our place, we have expertise in this part. Our forte is synthesizing nanoparticle right from 5 nanometer to 100 nanometer monodisperse. When I say monodisperse, the distribution size is around 10 nanometer to 10.1 to 9.9 nanometer. There is a high distribution we get now synthesize the nanoparticle for the application. And we do have registered recently granted a patent PCT on Alzheimer's disease using the nanoparticles. So we have got now total 14 patents. Out of that, 4 have been granted, 2 are PCTs. So now we are putting in the market this part of things. So I welcome you all to the universities. And thank you very much, Professor Ramchand and whole his team for giving an opportunity to be with you. Thank you very much.
Thank you for the informative speech, sir. Dr. TP Singh is ready with it, is ready online to share a few words with us. Sorry, we understand there's a technical glitch from Dr. TP Singh's side. I welcome our guest of honor, Sri Abhay Kumar Srimal, founder and chairman, Nano Life. Mr. S. Abhay Kumar is internationally renowned entrepreneur, known for his contributions in drug discovery and approvals of over 30 pharmaceuticals by regulatory agencies. Mr. S. Abhay Kumar, being a chemical engineering background, brings over 30 years of experience in the field of pharmaceuticals, Shasan Pharma. May I now request our guest of honor, Sri Abhay Kumar Srimal, to deliver a speech.
Respected dignitaries on the dais, I'm honored to share the dais with Professor Ramaswamy. Professor Ramaswamy ji is a legend in science and technology, and sitting next to him is a honor by myself. Thank you very much, sir. and other dignitaries on the dais, of the dais, and online. We have a lot of good speakers, and an old good friend of mine, Dr. Ajay Kapoor, also joined. I've been hearing that Dr. Ramsamy talks about application of nanotechnology in the marketing. How difficult it is in the marketing is something which I have experienced myself. And Professor Upadhyay rightly said that the Gujaratis always think in terms of research to rupees. And I don't know, being a Marwadi myself, what he will say. Well, I'll be converting that research into rupees or not. And being a Marwadi, I ensured that, I don't waste my time doing the basic research of producing nanotechnology particles. But we had the advantage of working with University of Missouri right from 2007. And in 2007, we bought their patent rights, worldwide patent rights for gold, silver, iron, and platinum nanoparticles to be made through green nanotechnology. We don't use any chemicals or solvents in the process of making green nanotechnology products. So for me, getting the basic research was already done. Now, application of this nanoparticles into the commercial application was my target. And from 2007, Dr. Ramasamy, we worked with University of Missouri, Dr. Kati Katesh, the scientist who really is called as the father of green nanotechnology. And we had worked with them to see that whether we can use radio-activated gold nanoparticles for treating cancer. And we did this, and we did an animal study also. We found that the cancer is completely eradicated, but it leaves the radioactivity in the body. So that was a issue we had, and we had to overcome this radioactive material being left in the body. And we said that how can we overcome this challenge? And that is where we used natural materials to convert gold and silver into nanoparticles. So, I mean, naturally, it's a patented technology, so I can't reveal more about the technology. But what I can tell you is that we started using its applications. The applications I see are huge, and I can tell you that we are probably the first company in India to have the silver nanoparticles approved by the food FSSAI as a preservative and a food additive. And I see a huge application there because the amount of vegetables, fruits, grains, and others which get decayed and get wasted can be saved by using these silver nanoparticles as a spray or a fogging material or any other application which can be done. So we are also working on gold nanoparticles on the cosmetic side and the iron nanoparticles. So the biggest challenge India faces is the anemia. And there is a complete anemia eradication program by the government because the iron oxide or whatever iron salts which are given for treating anemia gets absorbed not less than more than 5 to 8% of the body of the material. The rest are all going out. So if the nanoparticles can be used, I'm telling you, I think it can bring in a revolution. So this can be considered by the scientists here to see, and we are also working on it to see whether we can use this for eradication of anemia in the country. Besides that, on the biological application, sir, we started stem cell preservation in 2004. And after that, we started collecting the cord lining also. But right now, we are collecting the amniotic fluid also, which is around the baby. And this amniotic fluid, we bring it to the research center, processing center, convert that, decontaminate that, then convert into nanoparticles and make a patch out of it. And it has already been approved for commercial application. And there is a huge demand by the military and the other users where the wound care is is an issue. So this has proved to be a very successful product, and we are not able to meet the demand of the country or the world because the maximum amniotic fluid is produced here in this country, where we have the maximum number of babies. Uh, the application of gold nanoparticles has to gold, silver, or iron or platinum. Even the platinum nanoparticles are one area where I would definitely like to have a discussion with Professor Ramsamy separately, is to use these nanoparticles of platinum in the catalytic reactions. Why not we use, why do we use such large quantities of platinum where we can replace that with nanoparticles of platinum and not even go for recovery also? You don't have to go for recovery because the quantity which will be used will be minuscule. And this, we are working on some of the experiments which we can try and see whether the nanoparticles of platinum, palladium can also be used as a catalyst in the chemical reactions. The future is predicted to be of nanoparticles, and I'm sure the Mag Genome under the leadership of Dr. Ramchandran and others is doing a phenomenal job. We'll be happy to collaborate with you and work with you, sir, and the university, Meenakshi Sundaram, sir, and Ramasamy ji. We need your guidance on how to take these nanoparticles into the commercial application and make it useful for the society. Because finally, the idea of using any technology to see that it brings more effective results, but at the same time is the most economical. And under the nano mission, sir, we are the probably, I don't know about Ramchandra should have applied for some of the applications in the drug license. So as a drug application, we have applied for silver nanoparticles using green nanotechnology as an API. So we have to apply for the license for the API to the drug department. And we have already applied that, and hopefully we'll be able to see that light. Because once it is approved as an API, the amount of broad spectrum it opens up for marketing into antimicrobial agent, as an antimicrobial agent, will be a phenomenal application, replacing the chemicals and the biologicals or the bacteria or the others which are being used in the treating the current problems. We have also tried silver nanoparticles uh, for treating HIV in some of the cases and tuberculosis. I think these are some of the applications which are going to be huge applications. And once we get the approval as an API for silver nanoparticles, our next attempt would be to start the clinical trials and establish this as a drug. And silver nanoparticles is the most potent antibiotic known. And sir, the biggest problem the world faces today is that they don't have the next generation of antibiotics coming in the field. And this can be a drug, an antibiotic, which can be non-drug resistant antibiotic. And this, if it is approved as an antibiotic, actually in America, sir, the clinical trials approval, they straight away give you, after the toxicology studies and all, which we have already done. We have done extreme toxicology studies uh, uh, and proven that even at the high doses of silver nanoparticles is given to the body, it doesn't actually create any toxicology. See, there is a difference. People don't understand the difference between uh, silver nanoparticles and silver ions. So silver ions, people think that, it can be working as a nanoparticle. But silver ions, actually, when you start taking, it affects the body and the color of the body also changes. As Ramaswamy ji rightly pointed out, the properties of the nanoparticles completely change once they become nanoparticles. Like the gold, which you see it as a yellow gold, becomes completely like a red wine color when it is in nanoparticles. And silver becomes yellow. Silver becomes a liquid, light yellow color, which is white in color. When it is nanoparticles, it turns out to be a yellow in color. Similarly, iron nanoparticles. So they all have a different dimension, different properties. But as they rightly said, surface area becomes so big that it is able to at a very minuscule level, it is able to do a fantastic result. So I hope this one-day conference really brings out a lot of like, scientific advantages and advantages to the human being in the applications of nanotechnology in biologics. Thank you very much.
Thank you for sharing your thoughts, sir. I now request Dr. T.P. Singh to share few words.
You hear me now?
Yes, we can hear you. Yes, sir, please go ahead.
So, okay, thank you so much. I think I realized that it's better to go physically than online. And I think it's the best to speak at the end because everybody has been said about, everything has been said about everything. So anyway, I thank Priyanka and Ramchand for asking me to be part of this. So I think I said if Ramchand had told me before that Dr. Ramasamy is also part of this, I would have come physically. As he said that nanoscience is very old, and I think it's only nanotechnology which is now making a difference. We all have been doing this nanoscience when we talk about molecular science. So, but it's very, I'm very happy to see a lot of good things. As I think the combination of this nanoscience and nanotechnology is making a lot of difference in terms of applications. I only want to say that I'm a big admirer of these kind of efforts. I think Dr. Ramchand and Sam Santosh together, very good. They contributed very nicely. And I was very happy to hear also what Dr. Upadhyay said about his university. So currently, I think the environment in our country is such that lots of good things are happening. So we need to, I think there's one more interesting thing what you say about this autoimmune disease. There are lots of new ideas now how to tackle at molecular level also. So eventually, I think as said, in present time, the real applications are in biology and medicine. And this is an era of biological sciences. We are at a stage now that some knowledge we will gain, but we need to gain much more knowledge to tackle all these problems. And the time will come when we know everything about biomolecules, lots of issues can be sorted out very quickly. It's only the time now that we are working out molecular level to understand the processes, and these processes eventually become very important. You know, there's one more thing that we should realize. We haven't talked much about innate applications of the innate immune system, the molecules which are involved in the innate immunity. And that becomes important. This the other kind of problems which arise in terms of innate this autoimmune disease and other problems related to antibodies and so on will have less requirement. So I really I don't want to take much time because I think their time is already overshot. But I greatly appreciate the contribution of the companies run by Sam Santosh and Dr. Ramchand. And I was very happy to hear things from Dr. Abhay Kumar ji also. So I feel things looking very positive. And it's very nice to see that Dr. Ramasamy is part of this. I would say that I think it's a great opportunity that he's around in Chennai, and the kind of knowledge he has it's very, it can guide a great deal. I think talking to him scientifically is extraordinary. So all this setup today, I feel very happy and excited to see. and I wish you all the best. I don't want to say much because so many nice things I heard from so many speakers, and I see a lot of positive environment, and I'm very happy on this. So I wish you all the best. Thank you so much. Anil.
Yeah, thank you, Professor T.P. Singh. Great that we are missing you over here.
I think I should have been there, but anyway, it was very short notice, that's why. But it's very
Yeah, I
nice
know.
to see you doing well. I'm so happy on this.
So there is a slight change in program. What we are planning to do is that, Mr. Abhayji has to go. So he has another meeting to attend. But I was very particular that he should launch one of our products. We have a series of launches are there. So one of the launches which Kamalika was to do at a later period. So may I request Kamalika, please tell what exactly is the product she is interested to launch. And I would request Abhayji to launch that one. And she will be able to talk about the product at a later period. Then subsequent to that, we can have our tea and other talks and programs.
Hello all, good morning. So I'm Kamalika Roy Chaudhary. I work as a scientist at Maximum Technologies Private Limited. I take this opportunity to request Sri Abhayji to kindly launch some of our nanoparticles. So we have started with the bare iron oxide nanoparticle, as you have heard, and now we are expanding for other nanoparticles, including gold nanoparticles, silver nanoparticles, and differently coated various functionalized iron nanoparticles also. So if Abhayji can just launch some of our products, and later we can go ahead with the presentation. Okay, thank you.
Thank you all. I thank all the dignitaries for gracing the occasion, and I request all of them to kindly accept the memento. I request our chief guest, Padma Bhushan Dr. T. Ramasamy, to kindly accept the memento. Guest of honor, Sri Abhay Kumar Sri Srimal, and Dr. R.B. Upadhyay to kindly accept the memento. I request our patron, Dr. S. Meenakshi Sundaram sir, to kindly accept the memento. I request our advisor, Dr. C. N. Ramchand, to kindly accept the memento. Thank you all for joining us here. I request everyone to break for tea and join us here for the technical session that will happen around 12 o'clock. I repeat, I repeat, kindly join in the hall at 12 o'clock after your tea break. Kind attention audience, kindly occupy the rows in the front. Please don't leave them abandoned. Thank you.
Hello? Hello? Yeah, so far so good. Hello?
It's coming picture in picture what? No. Hello? Yeah.
Yes, we can hear.
Okay. Yeah.
We can hear you, please. Can you share your screen, your
Yeah,
presentation,
I have
please?
already shared.
Yeah.
So can you see my presentation?
Yes, please put on full screen.
Yeah, I have put it on full
Yeah,
screen.
so it's fine. It's going fine.
Okay. Thank you so much. Thank you. Sorry for bothering.
Just wait for a minute.
Is it the slides are changing?
Yeah. So I think now you will be just after the break, yours will be there. So like I think let us be like this. You can
Okay.
stop
Okay,
sharing,
fine.
be here. So when it starts, we'll just tell you. You can start
Oh,
presenting.
sure,
The audience
sure,
comes.
sure,
So we
sure.
don't have to reconnect back. So that's for that reason.
Okay, right.
Thank you. Thank you.
Okay,
Thank you.
thank you. Thank you very much. It's not looking at the president.
Hello, Sangeeta ma'am.
Yes.
Yeah, just
Yes.
we are also checking the next speaker. So we will revoke the presentation right from you. Give it back to you in about a couple of minutes.
Yes.
Okay, sure.
Thanks.
Okay, okay.
Hello, hello. Am I audible? Okay. Yeah, but video it is the main problem is it is video is not getting activated here. So can you see the full? Can you see the entire screen? My screen visible? Am I audible?
You are audible. Can you?
Is my screen visible? Can you see my
Yes, yes, yes.
We can see your screen, but
okay.
not your audio.
So
Can
in
you
the
share?
screen, yeah, just a minute. In the screen, can you see this portion? The video here it is showing it is not it cannot be activated.
No. Actually, he has shared this.
So now my presentation
Yes,
is visible.
presentation has come. Put it
Okay.
in full screen. Presentation has come. Yes, presentation has come, but your video has not.
Yeah, so the slide is changing, right?
Yes,
Am
yes,
I?
absolutely.
Okay.
Slide is fine.
Okay. So then we can go without the video, no problem.
No, but video, what it is showing?
You can see over here the video, it is not activated, means I cannot click on this.
Option ghost হয়ে আছে. Okay. Okay. Just give us 1 minute. We'll just try to figure it out. Otherwise, video presentation দেখা যাচ্ছে. That's not a problem.
Okay. Then it's fine.
Okay. 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 Fine. ঠিক আছে. Then can we just revoke your this thing presentation mode?
During my slot, then we can.
Yes, yes, sure.
Next in line.
I'm
After.
online. In the meantime, I'm online, so no problem.
Okay. Yes, yes. Dr. Shongita Rai is there first and then followed by you.
Okay, thank you.
Okay, fine. I'll give you a text. Okay. They need to over the host because we're running late. So I'll just pick up will come first. ये है वीडियो डिवाइस में
Sangeeta Roy, ma'am, are you there?
It sounds fine.
I'm privileged to invite Dr. Sangeeta Roy, Scientist E, Associate Professor, Institute of Nanoscience and Technology, Mohali, India, to be our first speaker. She is currently serving as the head of the Chemical Biology Unit of the Institute. She has published several papers in highly reputed international journals with an H-index of 26. Her current research is focusing on developing new strategies using novel peptide-based biomaterials and cells from diverse origin for tissue repair and regeneration. She'll be joining us online. I request you to address the gathering, ma'am.
Good afternoon, all. Let me.
Yes, you're audible. You're audible.
Yeah, sure. Thank you. So let me go to the screen. Yeah, so is the presentation visible?
Yes, ma'am.
Okay, thank you so much for your kind introduction. So So thank you. I would like to first thank the organizers, especially Dr. Kamalika and Dr. Priyanka, for giving me the opportunity to share our research with you today at this very crucial platform. So as you can see from the title of my talk, that our work focuses on minimalist peptide nanotechnology that are targeted for healthcare applications. So obvious question comes, what is the strategy we basically use? So basically, we use a classical strategy that is used by nature, which call it molecular self-assembly. So if you go into nature, you see plenty of examples of self-assembled structures in nature, starting from DNA double helix to collagen triple helix, then actin filaments, microtubules, many such structures, which does very important functions of our biological system. So if we closely look into the natural processes, we will see that there are three fundamental processes running hand to hand. So one is molecular self-assembly, biocatalysis, as well as the molecular recognition, which makes these complex hierarchical biological structures. So that does all the important life processes in our body. So now, if we go into the nature, so nature deals with all the big macromolecules, but we are not smart as nature is. So, learning from nature, what we are trying to mimic the similar complex structure from very small molecules, so that it can does a similar function. So, what is our choice of molecules? So, we basically take like this molecule where this is a kind of amphiphile, as you can see from the cartoon diagram. So we have a short peptide chain, which is no longer than two. It's usually 5 amino acid sequence, and at the N-terminal end of this peptide, it is capped with a large aromatic group. So you can see now it becomes a nice amphiphile. So when we put these molecules in water, so they actually start interacting with each other through non-covalent interactions like hydrogen bonding, pi-pi stacking, then anionic interactions, as well as hydrophobic interaction, and thus immobilizes the solvent, that is water basically, so to make this kind of self-supporting hydrogens. And if we really see into the picture, that it's like 99.9 to 99.5% is water. There's very small amount of your peptide is usually is needed to make this kind of self-assembled nanoscale structure. So our lab is basically working on these three overlapping domains. So first we identify from the nature the short minimalist bioactive sequence from the protein of our interest. Then we make hydrogen scaffold out of it. So to get the new functions, we always, you can imagine the traditional approach would be that you make a new molecule and you get it assembled in a new structure. So obviously it will give different functions, new functions. But what we try, we don't really want to make too many new molecules. Rather, we want to explore differential self-assembly pathways so that we can tune the physical properties of the nanoscale structure and the functions. So ultimately, our objective is to apply this hydrogen scaffold for interfacing, for making a good interface with the cell so that it can find future applications in tissue regeneration. So to start with, the basic sequence we have taken is the simple dipeptide sequence from the amyloid beta protein, basically. So you can see that this diphenyl sequence is at the N-terminal end is capped with a small aromatic group. So we have taken this molecule in phosphate buffer, and we have used the same concentration but applied two different methods for making them self-assemble. And you can see one of these method is heat cool, another is sonication, and through both this method they make nice gels. And if you closely look into the structure of the gels, ideally they should be similar, same molecule taken at same concentration. But you see, one is a transparent gel, the top one, and the bottom one is quite opaque gel. So if you see morphology at the nanoscale, you see a very distinct difference. So one is forming very nice nanofibrillar structure, another is forming twisted ribbon. So if you analyze the physical properties, you can see that the gels that are formed by heat cool method that are forming nice nanofibrous network are much stable. So they are melting at high temperature compared to the twisted ribbon ones. And if we analyze the stiffness of these gels, you can see the black bar. If you see that, you see that gel 1 is showing quite higher mechanical stiffness of these gels compared to that of the gel 2. So but we have, again, I want to remind you, we have taken the same molecule at the same concentration. So ideally they should show up similar properties. So what is basically happening, in these two different recipes. So what we imagine is that, or hypothesize that, by sonication, we are basically putting less amount of energy into the system. So molecule is probably going into the kinetically trapped structures. But when through heat cool, we are putting more energy through heating the system, and we are reaching up to the thermodynamic minimum possibly. So in order to find whether our hypothesis is right or wrong, so we have what we have done. We have taken both the gels, one is twisted ribbons, another is fibers, and then we have melted both of them at high temperature. And we see that after we cool them, so we can see both are forming nice nanofibrous network. And in order to find out whether the physical properties remain the similarly affected or not, so we have checked that the gel one, the black bird again, and gel to the blackbird, which were showing a quite distinct difference at the beginning because of their differential recipe. Now when we put the same amount of energy into the system, they are gaining, after 24 hours they are reaching up to the similar mechanical stiffness. So that says that with input of more energy, we are transforming the kinetically trapped structures now into the thermodynamically controlled product. So another interesting aspects of these gels, since we are going to apply them for future in tissue engineering applications, so it's important that they should be injectable so that cells can be entrapped within this hydrogel network. So that's why we have put stress on these gels, and we saw that the fibers are breaking down into shorter pieces and the gel is basically melted. So we are overcoming the weak intermolecular non-covalent interactions. So once we check up the stress out, we see, since these interactions are reversible interactions, so you can see that gels are regaining this fibrous network and similarly its strength over the stress strength cycle. So that says that we can easily entrap ourselves within these gels, which are injectable in nature. So to find out, as I said, that our approach is to control self-assembly pathways rather than making too many molecules, new molecules. So we want to see now, from the same molecule at the same concentration, can we create differential cellular response or not? How they behave with the cells, the differential nanostructure, although they have the same chemical functionality at the surface. So when we saw that, we can see that irrespective of the cell line, be it normal cell line, fibroblast cells, or it's a cancer cell line, you can see the red bars, that is a gel 1, heat cool gels, are showing up much better compatibility compared to the blue ones, that is gel 2, that is twisted ribbon structure. So if we see the cellular proliferation of the fibroblast cells, also we can see for gel 1 on the surface, cells are increasing in number over the time. And if we go to the analysis of the through the facts quantitative analysis, you can see that there is a 40% cell death is found in case of twisted ribbons compared to that of gel 1. So it's only more than 92% cells are viable on the nanofibrous network compared to that of gel 2, that is forming twisted ribbons. And it's very similar for the gel 1, it's very similar to that of the control. So that says our scaffolds, if it's a nanofibrous scaffold, that is forming very nice interface with the cells, and it is supporting cellular adhesion, growth, and proliferation. So now you can see the beauty of this work is that, so we use the same molecule and it creates, because of the differential recipe, differential nanostructure, which can create differential functions. So I have just given you an example where we have taken this amyloid inspiration from amyloid peptides. So here I will straight away come into the, for interfacial interactions with the cells, we have to consider the extracellular matrix of a cell. So if you go into the closely look into the extracellular matrix, so there is one of the functional protein that is present in the basement membrane is this protein, laminin protein. It's a large protein, 900 kilodalton protein. As I said that we are not so smart to handle this large protein. So we took a cues or inspiration from nature, and we found that there are two specific sequence, pentapeptide sequence, one is IKV, another is YIG. So these two sequences are functional sequence of this large 900 kilodalton protein. So can we make the similar structures through the formation of a hydrogen so that it can perform similar functions of that large protein. So in order to do that, what we have done, we have modified at the end terminal end with a FMOC group, and we see that both of them forms nice nanofibrous network. And when they're mixed together as that is there in the natural laminin protein, so we see a self-sorted network. You can see shorter fibers formed by more hydrophobic IKVAV on the top, and at the bottom you can see that the more hydrophilic sequence is forming nice nanofibrillary network. And if you go into the analysis of this strength of these materials, you can see the composite gel and enhancement in strength is being observed. So now we want to see that how they work with the cells. So since this laminin has a connections with the neuronal cells, so we thought of culturing, putting neuronal cells on this surface of these gels, and we took C6 glial cells, and we saw that the scaffolds are quite compatible with the cells. And over the time, you can see that over 5 days on the composite gels, actually, we found that the extent of cellular proliferation is highest. And once we do the live date staining, and we found that cells are quite happy on these newly designed surfaces. Again, I want to point out here, we have taken the cues from that large protein, and basically we are only confined into the pentapeptide sequence, which is very short. Again, it's confirming that by adapting a minimalist approach, we can create a structure which is basically mimicking this the structure of this large protein at the nanoscale. And then we wanted to look into the other performance of these cells on this scaffold also, and you can see that our scaffold is promoting the neurite extension to a great extent compared to that of the control cells. And also they are maintaining very nice beta-3 tubulin expression, and there is no cell cycle arrest is there. So it says that the cells are quite comfortable with this newly developed minimalist scaffold. So moving on further, so what we see is that if you look into again the extracellular matrix, so laminin is one of the functional protein. So there are a lot of other proteins that are also present. One of the major part of this extracellular matrix is composed of collagen, so which is having a basic repeating unit of glycine, proline, and hydroxyproline. So what we have done here, we have mutated this proline, and we have put a serine residue here, which forms in water nice spherical aggregates, but it doesn't form a entangled network of fibers. So we have now mixed, as it is there in the natural system, with the 10% of the laminin mimetic peptides, this IKVAV and BioIGSR, and we can see that nice network of fibers have been formed. So now when we want to culture the cells on the surface, so what we have seen, because of the dual functionality of collagen and laminin, now the scaffold is more versatile. So you can see that because of the laminin, the C6 cells, those are glial cells of neuronal origin. So you can see that the scaffold is supporting the neuronal cells as well as they can support the fibroblast cells, which are in connective tissues. So since we have collagen in the collagen functionality into the system, again, you can see from that large protein, we have taken this only one tripeptide unit, and we have mutated it and capped at the N-terminal end with the aromatic group. And we can see from the 2D culture, cells are quite happy on these scaffolds. So moving on further, so I'll just show you now some of the glimpses of some of our other work. So that's like one of the very crucial protein is cadherin protein. So it mediates basically the intercellular adhesions and degradate differentiation and morphogenesis. So when we look into the literature, we didn't see any of these short peptide sequence based gels are reported. And what we thought that why don't we consider this protein to go ahead to find out whether we can create a suitable scaffold for the cells, and we succeeded onto that. We have taken only this pentapeptide sequence that is HAVDI, and we capped it at the N-terminal end with aromatic group, and it forms very nice fibrous network, and it's supporting both it's quite versatile. It's supporting both the cells of neuronal origin as well as fibroblast cells. And we have additionally saw that the scaffolds are promoting the cellular migrations, which are very crucial if we deal with the wound healing applications of with this kind of scaffolds. So moving on further, till now I have shown you the promises of our peptide scaffold, how they are creating a very suitable interface with the cells. Now extracellular matrix again is composed of other entities, like there are sugars, polysaccharides are there. So what we thought, and if we look into the peptide gels, we have seen that the gels have the mechanical stiffness which are not very high. So for long-term usage, that might create limitations for us. So what we thought, that can we make a composite gels with any of the polysaccharides or natural polymers like cellulose? So what we have seen, so basically we have taken the biomass derived cellulose and we have done a tempo oxidation, and we end up with the nanofibrous cellulose. And we saw that when we mix our peptide with this cellular scaffold, you can see the peptide chain, the green one, it's showing quite less strength. By incorporating peptides into the cellulosic scaffold, we can see the stiffness of this gel has been increasing. So that way we can impart more stability into the cells. Now we want to understand that whether this composite scaffold also supporting the cellular adhesion and proliferation. So you can see very nicely over the time, this is the peptide alone. So there is proliferation. The extent of proliferation is less, but the extent of proliferation is more when you have a composite scaffold. This one is indicating that this is a composite scaffold, 10 is to 1 peptide is to cellulose ratio. And one interesting thing is that the previous one examples I have shown that when we cultured the cells on the surface of this nanofibrous scaffold, this is, we have entrapped the cells within this scaffold in a 3D environment, the way we have in our biological system. And we saw that, so you see a little less green cells in case of peptides, while you see more extent on the cellulose, more extent of green dots with the cellulose itself as well as in the composite gels. And you can see very nicely our composite gels are basically performing in a similar way to that great extent as that of the commercially available scaffold people are using, the matri gel. And once we,
Dr. Roy, excuse me. Yeah,
yeah.
if you can just conclude within 1 2 minutes. We
Okay,
are
sure.
running
I will do
with
that. I have
deadline.
one more slide, so I'll go fast. Yeah, so we can see that this doesn't evoke any inflammatory response as well. So this is another example where we have used another neuronal protein, that is tenascin C. So again, we have derived this octopeptide sequence from there, and we made a suitable scaffold, and that is injectable in nature. So moving on further, the final one I would like to highlight here is that here we have made a composite scaffold which are inspired from another small molecules of the native HCM system. Is the heparin or heparin sulfate, which is basically a proteoglycan. So that's why we make a composite scaffold from it, and we can see the cells are quite happy in the 3D culture condition and doesn't evoke a significant response, inflammatory response as well. So that says the beauty of this nanofibrous scaffold. So I would like to wrap up here. So I hope I have been able to convince you that taking the cues from natural system, how we can make use our minimalist approach to make new scaffolds which are suitable for the cells and can perform the similar functions to that of the large protein. And by changing the the physical uh, just the self-assembling pathways, how we can make new nanoscale structures which perform different functions. So at the end, I would like to thank all my students who did really hard work to make all these uh, results come up, and my collaborators, the former students, undergraduate students, and the funding agency, of course. And I would like to thank the organizers again, Dr. Priyanka and Dr. Kamalika, for giving me the opportunity to share our work with you. Thank you very much for your kind attention. I would be happy to take any questions if there. Thank you.
Thank you for the informative speech, ma'am. We'll be having the query session later. The attendees are requested to post their questions in the chat box. The
Right.
questions will be answered.
Okay, thank you.
I'm proud to invite our next speaker, Dr. Saugata Ghosh, visiting faculty at Kosakat University, Bangkok,
who is also Associate Professor in the Department of Microbiology, School of Science, RK University, India. He is former visiting professor at the Department of Chemical Engineering, Northeastern University, Boston, USA. His areas of research include nanomedicine, drug discovery against infectious diseases, diabetes, oxidative stress, and tumors. Having expertise in nanomaterials and nanobiotechnology, he has 207 publications on record. He'll be joining us online. I request you to address the audience, sir.
Hi, Dr. Saukata. Can you hear us? I think you have logged in with two systems. Dr. Saukata, I think you have logged in with two systems. Can you just log out one? Hi, Dr. Saukata.
Yeah, am I audible?
Yes, yes, you are audible now.
Okay, great.
Can you just share your screen?
Yes. I'm sure.
Yes, fine. Yes, please go ahead.
First of all, I thank the organizers of Icon Bio 2023. And as per the topic that is today we are discussing, it is the how we can develop some products from the nanotechnology. What is the commercial potential of nanotechnology? So here I will be focusing mostly towards the type of nanoparticles we are generating and how it can actually help in the healthcare section. So as we know, like there are different methods for synthesis of nanoparticles, and mostly the two approaches, that is the top-down approach and the bottom-up approaches, are there. So for our studies, for our researches, we are mostly taking up this bottom-up approach, where first very small nuclei are generated, from that bigger particles, and then we get nanoparticles, which are in a size range of 1 to 100 nanometers. Now, what we are doing exactly, what kind of studies we are doing in our lab, we are trying to see or mimic different nanoparticles that are found in the nature and how we can use natural resources for our nanoparticles. So if you're looking at this butterfly, you can see there are different colors. That is the black color, light brown, deep dark brown, and blue colors in the wings of the butterfly. But actually, these wings are, or these colors, the wings of the butterflies are not colored. They have nanotextures. These nanotextures give them the color. These brown colored parts of the butterfly, these are called androconial organs, which are the male pheromone producing organs. From these, they produce pheromones to attract the female butterflies. So if you are looking at these dark brown structures, you can see that nanotextures over here is different and the scales are different over here. Similarly, the light brown, the black, and the blue patterns. So that means it is not only that the nanoparticles are present, means we can synthesize them only in the lab, but the nanoparticles or nanostructures or the nano patterns are present in the nature also. So we are trying to develop different types of means nanomedicines out of this, and we know that these nanostructures have so many different types of applications. Now, in our lab, we have synthesized these nanoparticles. Like the first one I'm showing over here, this is the gold nanoparticles, which are spheres, hexagons, and triangles. This is the silver nanoparticles of different sizes. This is gold silver bimetallic nanoparticles with a gold core and silver shell. So this helps to kill the leishmania as well as eradication of the bacterial biofilms, which are mostly responsible for different infections. The fourth one is the copper nanoparticles, which are anti-diabetic in nature, and they can inhibit different carbohydrate metabolic enzymes. Here we have the magnetic nanoparticles with saponin conjugation on their surface. And here we have another magnetic nanoparticles with curcumin conjugation. So I'll be telling you the potential of these two nanoparticles for today's talk. Apart from that, we have gold, we have polymers and the liposome formulation for anti-cancer drug delivery. We have platinum and palladium nanoparticles and also defect-rich zinc oxide nanoparticles for anti-diabetic as well as antioxidant nanoparticles. So all these nanoparticles which we have developed in the lab, they have different types of or multivariate therapeutic potential. And our researchers are generally funded by the DVT, CSIR, and DST also. So why or from where are we getting inspiration to develop these nanostructures, which are mostly derived from the natural resources? So if you are looking at these potato-like structures, these are called air potato or Dioscorea bulbifera. Now, this Dioscorea bulbifera, it is a climber and it is potato-like structure which has so many different therapeutic applications. One single bulb will have so many different therapeutic applications. So this plant generally grows like a climber, and the growth rate you can see 15 centimeter per day. The growth rate is so high. In the month of June, July, there is bulb-like structures. In the month of September, there is flower, and in November, these bulb-like structures they fall on the ground as there is a dieback mechanism. The plant dies means if you apply more water, fertilizer, or whatever you supply to the plant, but still it would die in the month of November. And what remains on the ground is these air potatoes. So we generally use these air potatoes or these Dioscorea to synthesize different types of nanoparticles. Why? Because if you're looking at the chemical and physical synthesis methods, then we use different types of reducing agents and capping agents, which are generally chemical in nature, which are hazardous, corrosive, and it has lots of disadvantages while developing nanomedicine. Now, this plant, the Dioscorea bulbifera, this air potato, it is rich in phenolics and flavonoids. It has ascorbic acid, citric acid, starch, and saponin. So all these chemicals, they not only act as very strong reducing agent, which can convert the metal ions to their respective nanoparticles, but also these starch and saponins, they can help in the shape evolution and they give stability. Since we are not using any kind of chemical entities over here, these nanoparticles are biocompatible, and as we see that the same plant can produce different types of nanoparticles, we can develop nanoalloys and multi-metal nanoparticles. So it can have more enhancement of the activity. Now coming back to cancer nanomedicine, we know there are different types of drugs like cisplatin, carboplatin, oxaliplatin, and all this. So these drugs, when you are applying to different types of cancer, the drugs does know that which one is the body's healthy cell and which one is the cancerous cell. So it can go and indifferently kill many healthy cells also, and that's why we have lots of side effects to cancer chemotherapy. So in order to address this, the nanoparticle that we develop in our lab is the magnetic nanoparticle, and these magnetic nanoparticles are functionalized with saponins from Dioscorea bulbifera, that is diosgenin. Now for functionalizing, we used a linker, and this linker is citric acid. So here you can see it is magnetic nanoparticles. So as we know, iron is also present in hemoglobin, so it is compatible. Citric acid, we know in Krebs cycle, it is a means regular component of the cell, and diosgenin, it is a herbal derivative. So that's why all three components which are present in this nanomaterial or nanocomposite, these are all very, very biocompatible. Now, when we synthesize this, we checked it with the help of FESCM and HRTM, and we found that the nanoparticles were in a range of 12 to 21 nanometers. So it is very small. And why small is important? Because if the nanoparticles are lesser than 80 nanometers, they can be easily eliminated out from the body with the help of urinary system, to the kidneys, to the ureter. So that's why it is very important to have the nanoparticles very small. So here we can see the particle size analysis. The iron oxide nanoparticles are smaller, and these are the hydrodynamic size of the aggregates. Since these are magnetic, we do not have discrete individual nanoparticles. We have it in aggregates. So we have iron oxide nanoparticles which are around 65 nanometers. Then iron oxide citric acid capped nanoparticles which are around 120 nanometers, and diosgenin capped nanoparticles, diosgenin functionalized nanoparticles, which are around 150 nanometers. So how we confirm these? With the help of FTIR. So here the red color, you can see this is the free diosgenin, and the same peaks in FTIR we also find in the composite. So thus we confirm that okay, diosgenin is functionalized. So when you see the anti-proliferative activity against the MCF7, the breast cancer cell line, we can evidently, it is quite evident, quite clear that these diosgenin functionalized nanoparticles had more activity as compared to the free counterparts. Free citric acid 17%, free diosgenin 33%. But when we have the composite, it has 51% anti-cancer or anti-proliferative activity against the MC7. Now, one of the main important feature is the cancer cell in benign state. When it's benign, it's a tumor. In a tumor state, it is no problem. But when it starts metastasizing, it becomes malignant, and then the cancer moves. So the motility of the cancer or migration of the cancer cell is very important for its pathological implications. So that's why our aim was to see whether we could inhibit the migration of the cancer cells. So this is a lawn of MCF7 cells, and when these gaps are prepared, these gaps are produced over here, after 24 hours, we could see that from both sides, the cancer cells had migrated and the gaps were sealed. So there were more migration. Similarly, in case of iron oxide nanoparticles, lot of migration was seen. Iron oxide capped with citric acid, migration was little less. Here, when diosgenin was conjugated, then migration was very less. These are the dead cells we can see in the between, but migration was very less. Similarly, with only diosgenin also, it could prevent migration, but evidently with diosgenin composite, the migration was very less. So you can see how much was the migration. With iron oxide, the cellular migration was almost 90%, 89%. When we conjugated citric acid, cellular migration was 50%, and when diazoxide was conjugated, it was very, very less. It was minimum, that was 40% cell migration. We wanted to see whether the cells were getting inhibited by apoptosis or necrosis. So by dual staining, here we are using Alexa fluor, which stains the phosphatidylserine. Phosphatidylserine is externalized when the cells undergoes apoptosis, and PI, propydium iodide, enters. So here we can see there is no green color fluorescence, which is characteristic of phosphatidylserine, and neither there was PI. But when iron oxide with diazoxide was applied on the cells, we could clearly see that green fluorescence as well as red fluorescence. So that means phosphatidylserine was externalized and the cell membrane integrity was lost. So that's why we are getting apoptosis of the cells. Likewise, there is another entity that is called curcumin. So you can see curcumin has so many, it can inhibit so many different type of cancer. Why? A single molecule of curcumin, I repeat, a single molecule of curcumin can modulate so many different genes. So it can downregulate all these brown color cancer causing genes, and it can upregulate these blue colored anti-cancer genes. So that's why this curcumin is so important. But one of the most important problem is the bioavailability of the curcumin, because it is non-polar. It does not dissolve in water. So we conjugate curcumin via citric acid bridges to iron oxide nanoparticles, and these nanoparticles were around 15 to 20 nanometer. Again, these were very, very small nanoparticles. So we checked the agrobacterium mediated tumorigenic model in potato. So you can see around 80 to 90% tumor suppression was seen over here. So the brown color is because of the tumor, and when there is no brown color in presence of iodine, it becomes violet black in color. So here we can see that only citric acid there is tumor, but when it is with curcumin, the tumor was inhibited. At the same time, these nanoparticles were also able to scavenge the free radicals, because we know cancer is caused because of mostly ROS, reactive oxygen species, and oxidative stress. So this curcumin conjugated nanoparticle had highest free radical scavenging activity, almost 40% compared to the free counterparts. So conclusion, we can see that these magnetic nanoparticles, what MagGenome is actually dealing with, is actually very, very important and significant domain for developing nanomedicine. So these functionalized magnetic nanoparticles can be used not only for anti-cancer, but targeting the drugs to the anti-cancer site using an external magnetic field. Again, cancer hyperthermia, we can create heat in the localized heat in the cancer cells, and we can kill the cells. So that's why it is very, very significant. And we have published different books related to nanomedicine. So whatever research we are doing, whatever prospects are there about different nanoparticles, magnetic nanoparticles, biogenic nanoparticles, are published in this book. And finally, I thank Dr. Ramchand for giving me this opportunity, Dr. Kamalika for arranging and inviting me for this, and all organizers of Icon Bio. And I thank my funders and the PhD students and MSc students who were the integral part of this work that I showed over here. Because of time limitation, I wanted to stick to the time and I had to rush. So I apologize for that. Thank you so much.
Thank you for sharing your knowledge with us, sir. Once again, I request all the attendees to post your questions in the chat box. They'll be answered later. Our next speaker, Professor Deepak T. Nair, Professor, Regional Center for Biotechnology, India, is an Indian structural biologist known for his studies on DNA and RNA polymerases. He has obtained new insight regarding the molecular mechanisms that determine the fidelity of the replication process in bacteria and flaviviruses. His laboratory has shed new light on the strategy utilized by DNA polymerases to prevent ribonucleotide incorporation, and he has published more than 47 research articles in various national and international journals. He received the Shanti Swarup Bhatnagar Prize from CSIR for Science and Technology, one of the highest Indian science awards for his contributions to biological sciences in 2017. He'll be addressing us online. I request you to enlighten us with your words, sir.
Hello, can you hear me?
Yes, sir, you're audible.
Okay, thank you. Good afternoon, everybody. My name is Deepak Nair, and I am going to share my presentation now.
Sir, can you just share your screen?
Can you see?
Yes, it is visible. Can you make it a slideshow?
Yeah. Okay. Is it visible?
Can you make it a slideshow?
I have made it a slideshow.
We are able to see your presentation. And then can you make it a slideshow?
I have already made it a slideshow.
Okay, just go ahead.
Can you see it now?
Yes, sir, you can go ahead.
Can you see my cursor? Anyway, my name is Deepak Nair, and I am at the Regional Center for Biotechnology. I am a structural biologist, and I would like to thank Dr. Ramchand for inviting me for this occasion. Congratulations for the new product launch. And it's always been a pleasure interacting with Dr. Ramchand because of his generally very positive about many things. So I'll quickly start my talk. My talk is generally my lab uses structural biology techniques to look at molecules, enzymes involved in replication and repair. Years we have we have also started looking at therapeutic molecules, and recently we had the opportunity to work on one of them, which is a broadly neutralizing monoclonal antibody against SARS coronavirus 2. So as all of you are aware, this is the coronavirus on them, which are the ones, this protein is the one, the structure of which you can see here. It interacts with the ACE2 receptor and that is what allows the virus to enter the human infection. So this spike protein, you can see the domain organization here. It has what is known as the receptor binding domain. Interacts with the ACE2 receptor, and you have this receptor.
Excuse.
Yes. Yeah.
We are not able to see the slide change.
It's not changing here. I've been
Yes,
changing.
the slide is not changing.
Now what
Can
do
you
you
just
see?
check once?
Okay, what do you see now?
Now it is fine. Okay.
So when it is in slideshow mode, then it is not visible.
Now it is fine. You can go ahead.
Now can you see the slide?
Yes, sir. Yes, sir.
Okay. So I'll just continue in this manner only. Okay, so this is spike protein SARS corona 2 virus, and you have this which is the one which actually interacts with the ACE2 receptor on humans, and in that which forms most of the interactions with the ACE2 receptor. So we are at RCB, Regional Center for Biotechnology, and this is part of the NCR Biotech Science Cluster with and the neighboring institute is the Translational Health Science Technology Institute, where Dr. Rajesh Kalra was working, and he has recently moved to the Indian Institute of Technology. So his lab made this P4A2 antibody, monoclonal antibody, which is made using this spike RBD of the alpha strain of SARS coronavirus. And what they saw was that this P4A2 map can utilize different SARS coronavirus to variants of concern. And this is the data from the virus-induced cytopenic effect assay. And what he saw was that the P4A2 monoclonal antibody can bind to the spike RBD with very high affinity. So you can see that against the spike RBD from the reference strain as well as the strain used for immunization, alpha strain, the affinity is really, really very high. And it is substantially high in the nanomolar range for the other variants that were tested. So what we did was we took that the Fab fragment of the P4A2 molecule and made a complex with the spike RBD, and we got crystals of this. Work was done by Dr. Naveen Narayanan, who was a student in my lab and now is moved on to a postdoc in Canada. So we got crystals, and the X-ray diffraction data was collected at ES, European Synchrotron Radiation Facility, which is located in Grenoble in France. And this is what the diffraction frames look like. The structure was solved, and this is what the final refinement statistics look like. We have the structure to a resolution of 3 angstrom, and the structure has been 7W code. So this is what the structure looks like. Here you see the Fab fragment. The orange is the heavy chain, the cyan is the light chain, and in magenta you see the spike RBD molecule. So as most of you are aware, the Fab or the immunoglobulins bind to their cognate antigens through hypervariable loops, which can be seen here. So a paratope made from hypervariable loops, which can be seen here, and 3 hypervariable loops from light chain and 3 hypervariable loops from heavy chain contribute to the antigen binding site. So these are some of the. So this is the spike receptor binding motif that you see here, and some of the interactions which hold the uh, antigen in the antigen binding site are interactions between residues of the heavy chain and the RBM, the receptor binding motif of the spike RBD. Some of them are shown here. The most important One of the most important interactions is the fact that this F486, phenylalanine 486 residue is present in a hydrophobic cavity. So this picture shows you the surface of the antigen binding site on the antibody, and you see that there is a hole formed here into which the aromatic group of the phenylalanine 486 antibody is present. And this is a very strong interaction and very specific interaction. There are other interactions also, but this is a very important interaction which ensures that the P4A2 antibody binds to the receptor binding motif of the spike RBD with very, very high affinity. So what you see next is the deposited structure of the ACE2 receptor in complex with spike RBD. And the affinity of ACE2 for spike RBD is 44.2 nanomolar. So here what we saw is that some of the residues which are important for interacting with the spike, with the ACE2 receptor from spike RBD are also involved in interaction with the P4A2 antibody. So if you take the structure of the P4A2 antibody Fab fragment bound to spike RBD and you take the structure of the uh, spike RBD bound to ACE2 receptor and you overlay the spike RBD structures in these two complexes, you will get something like this. So you see that the ACE2 molecule is present where the Fab molecule is present. So if a Fab molecule binds to spike RBD, then the ACE2 molecule will not be able to bind. And by simple steric exclusion, the virus will not be able to latch on to the human receptor for infection. And that is how the neutralizing antibody works. So it is known that the spike protein exists in two conformations. One where the receptor binds conformation, and this is the conformation in which it can bind to ACE2 receptor. And we see, based on computational modeling, we see that the P4A2 antibody can bind to the spike RBD even in the up conformation as well as what is known as the down conformation. In this down conformation, the spike RBD actually cannot bind to the ACE2 receptor, but the P4A2 antibody fragment is able to bind to it. Now, the structure was with the alpha strain, spike RBD from the alpha strain, and using computational modeling, we made models of the structures with strains from different variants of from different spike RBD from different variants of concern like beta, gamma, delta, and kappa. And we see that whatever mutations are present in the receptor binding domain in these strains, none of them will interfere with binding to P4A2. And this was true not only for beta, gamma, delta, kappa, but also for Omicron BA.1, which has 15 mutations, but none of them will interfere with binding. When we were doing this work, and we saw that P4A2 can neutralize the Omicron BA.1 strain, as well as the P4A2 can bind to spike RBD from the Omicron BA.1 strain with affinity. And so overall, P4A2 can bind to spike RBD of most of the, meaning almost all the known variants of concern of SARS coronavirus at that stage. And experiments were done on animals to check the level of protection afforded by P4A2 against SARS coronavirus infection. So whenever there is SARS coronavirus infection, the weight of the animal drops, and if you have a therapeutic agent, it can prevent this drop in mass. So this is what you see here for different strains when they are treated with monoclonal antibody, their relative body mass, their body mass does not fall by much. So that shows that there is protection provided by P4A2 against deleterious effects of SARS coronavirus 2 infection. And this was supported by the data obtained from qPCR to detect viral RNA, where also you saw that the amount of RNA detected was a lot less compared to when there was no antibody. So experiments were also done to look at lung morphology, and it was seen that when you have uninfected, you have very nice morphology. When you have infected, you see that the lungs look really sick. But when you treat with different amounts of the antibody, and for different strains, you see that the lungs don't look as sick as when there is no antibody. So definitely there is therapeutic protection to lung morphology also when you use P4A2 antibody. So this is sotrovimab, which was a monoclonal antibody which was used for treatment of SARS coronavirus 2, and compared to sotrovimab, and it was seen that uh, it binds slightly better to the spike RBD of Omicron BA.1, and it is able to neutralize better than sotrovimab. So overall, the summary is that P4A2 provides protection against the tested VOCs, and this ability may be retained for majority of future. P4A2 mab can be used alone or in conjunction with other antibodies for as an effective prophylactic or therapeutic strategy against SARS coronavirus 2 infection. And this paper appeared in PLOS Pathogens. And this is a collaboration between five different institutes. So based on the structure that we have, we made models of spike RBD from bound to P4A2 fab, spike RBD from different Omicron lineages, and we see that for BA.2, BA.3, BA.2.75, and BA.2.212.1, the antibody should be able to bind very tightly and should be able to neutralize these Omicron lineages. However, some 6 BA.45, where that phenylalanine that I showed you has mutated to either serine or valine. You can, and this will lead to a drop in affinity. And if you look at the current prevalent lineages are all arise from this XBB, which has this now is using the structure engineer the paratope of P480 such that it will be able to bind to these new strains of uh, Omicron coronavirus 2 also. As I said before, all the work was done by Naveen Narayanan, and these are the other people in the lab who are looking at different molecules involved in replication and repair. I'd like to thank the Central Instrument and also the Advanced Technology Platform Center in RCB. All of you can note this website address down. People who run the audience, you can also access all the facilities which are present in the ATPC. It's on a payment basis, and there are 6 different facilities. You can, and if you want to access the resources here, please send the email to me. And for X-rays, I would like to thank the X-ray diffraction facility in RCB and also the beamline scientists at ID 30A1 of European Synchrotron Radiation Facility who helped us collect this X-ray diffraction data. This is part of a program where Indian researchers can access resources at facility, and this program is funded by DVT, and it has this program so that Indian researchers can continue accessing all the facilities at ESR. Thank you.
Thank you for those enlightening words, sir. I'm privileged to introduce our next speaker, Ms. Anu Varghese, scientist, Genome Technologies Private Limited, Kochi, who has experience of more than 8 years in biotechnology, genomics and molecular biology, and R&D experience in product development, DNA and RNA extraction using magnetic nanoparticles from various biological samples like saliva, animal tissue, plant, blood, and bacteria. She'll be joining us online shortly. I request you to address the gathering, ma'am.
Am I audible?
Yes, ma'am.
Yeah. Is the presentation visible now?
Yes, ma'am.
Is it in a presentation mode?
No, ma'am. I think you need to choose the launch option.
Anu, can you just click the launch option?
Yeah. Can you see now?
No, can you just switch the tab to your presentation?
Now, is it okay?
Yes, now it is fine. Kindly make it a slideshow. Oh, fine. Go ahead.
Yeah. Actually, good afternoon, everyone. So myself, Anu Varghese. So I'm working in Maximum Technologies R&D department in Cochin. So my work is regarding magnetic nanoparticles based total RNA extraction from plants. So whenever we talk about RNA extraction, RNA extraction is important in terms of gene expression, identifying the genetics variants, and crop improvement studies. And the extracted RNA has to be compatible with the downstream applications, and the downstream applications include RNA sequencing, that is NGS. Then it has to be downstream compatible with the real-time PCR, PCR. It has to be compatible with the cDNA library preparation, northern blotting, and cloning. When we talk about RNA, there are different types of RNA. The most important type, or the largest type, is ribosomal RNA, or rRNA, and that is the type of RNA that is visible in gel electrophoresis. And as we are talking about plant RNA, there are mainly three different types of rRNA. One is 18S, the smaller subunit, 28 is the larger subunit, and 5S and 5 point is subunits. Then we have messenger RNA, transfer RNA, small non-coding RNAs, microRNA, and small interfering RNA. As we are talking about plant samples, the major source for RNA extraction in the case of plant is leaf. Actually, it is because leaf is the easily available sample. So that is why leaf is considered to be the most important sample. But we can extract, because of the gene expression studies in a particular localized gene, to study the localized gene expression, we can extract RNA from seeds, floral parts, stem, root, then tissue culture, callus, and fruit kind of samples. So then, regarding the challenges that we face when we talk about plant RNA extraction is, since it is RNA extraction, ribonucleases are the biggest challenge that we face, because ribonucleases is a ubiquitous enzyme that cleaves RNA. Then next comes the polysaccharides, or the structural components of the plant, which can be like cellulose or starchy substances that is present in seeds. So this can interfere with the RNA extraction, or it can interfere with the downstream applications as well. Then the next category includes the secondary metabolites. Secondary metabolites in plants include polyphenols, flavonoids, or terpenoids kind of substances. And the peculiarity of the secondary metabolites is that when co-purified with RNA, it can also interfere with the RNA, downstream applications. Next is genomic DNA. Of course, it is since it is an RNA extraction, when genomic DNA comes, it is a contaminant as well. Then regarding the pigment and proteins, we know about the pigments that chlorophyll is one of the biggest, the pigment present in leaf. And there are some other pigments like anthocyanins, or kind of pigments that are present in the floral parts. So this can also interfere with the RNA extraction. Then next is the plant protein. So these were the challenges that we face in plant RNA extraction. Then different types of RNA extraction usually we have is, the first generation is actually phase separation, or the conventional method. In the image itself, it is very clear that we have three phases. It is actually, it will have a bottom organic phase. There will be an interphase where proteins are precipitated, and the aqueous phase, which will have the RNA. The problem with with this thing is like, when you transfer the aqueous phase, there are chances for the interphase or organic phase to be transferred to our the co-purification can occur. So it is always a laborious protocol with the increased amount of organic solvent carryover. Then the next generation comes the silica-based spin column, and this is the biggest category of RNA extraction kits. Majority of the commercial kits are silica-based spin column. The problem with this thing is like actually multiple centrifugation. When you take perform an RNA extraction using a spin column, it will have multiple centrifugation, and it can go up to 5 to 6 times of centrifugation for a single extraction. So multiple centrifugation means consumable. There will be more consumables to be used. So, and another thing is like, it has a limited binding capacity. That is, a particular amount of RNA could only be bound to the silica column, which cannot go beyond that. Then the third category, or the next generation, is actually magnetic nanoparticles or bead-based system. Usually, when we talk about magnetic nanoparticle, iron oxide nanoparticles are the largest, are the important type of nanoparticle used for nucleic acid extraction. The peculiarity is that it has an increased surface area. Since it is nanoparticles, it has increased surface area. So increased surface area means it can bind more amount of RNA than in the other two methods. And it is even more convenient. There are no phase separation or no organic solvent carryover or any intermittent centrifugation step. So it is always a convenient one. Then it has a higher binding capacities, and I have already mentioned that it is because of the increased surface area. And there is always a scope for automation when we use a magnetic nanoparticle based system. So the system what we developed was, actually, for every plant RNA extraction, there will be a sort of tissue homogenization which can happen using liquid nitrogen or any other bead beating techniques, which will be followed by a lysis step, and lysis which is happening at a higher temperature, followed by a precipitation to remove the contaminants. And during the binding step, the third step, during the binding and washing, nanoparticles are added to the plant lysate, which will selectively bind the RNA to the nanoparticles, while the contaminants will be removed in the supernatant itself. So the RNA bound to the magnetic nanoparticle could be washed, and later in the later stage it could be eluted as a pure RNA. So that is the protocol that we adopt for the we adopt here. So the highlights of the protocol is, as I've mentioned, it doesn't require any phase separation, just like any conventional method. And here there are not much use of hazardous organic solvents like phenol or chloroform. And the quality of the high quality and high yield, because high yield is because of the higher surface area. Next, it is actually the effective removal of contaminants, and the eluted RNA is downstream compatible. This is actually a representative data between the magnetic particle based system against the column base, and the column we selected here is actually a commercially available silica column one. Here we selected different plant samples like leaves, seedlings, seeds were selected. Here you can see that in the highlighted ones, the purity, we know that the purity profile of RNA should be 260 and 260 by 280 and 260 by 230 ratio should be above 2. But when you see that in the column-based system, the purity profiles were compromised in a lot of samples, while in the case of a coconut sample, you can see that even the yield was very much compromised. But when you compare it with the magnetic system, it is most of the cases we are getting good yield as compared to the silica column. So this is just a representative data. So this is actually a comparison data of 18 samples, and the samples are written on the are represented on the right of the right table. This is actually the total recovery, RNA recovery, that we are comparing against the magnetic particle. It was compared against the silica column one. You can see that in most of the cases, except in a few, 1 or 2 samples, in all other cases, magnetic bead-based system has got higher yield than the silica column based system. And in the first sample, that is mustard sample, it was even going up to 110 microgram from 100 milligram of tissue. So this is actually the gel image. Now here you can, in the highlighted ones, these are actually, the highlighted ones are actually the column-based ones where there was no RNA at all, while at the same time we were able to get good amount of RNA with the good amount of intact RNA from using our magnetic particle-based system. So this is actually the RIN profile. We know that for RNA analysis, RIN profile is the last word. Here also you can see that in some sample, on the left side it is paddy samples alone, the different tissue types of paddy like stem, seed, seedlings, etc. And on the right side it is different leaf samples of different species. And you can see that in certain samples like coconut and palm, there are no band in RIN profile also. That means RIN profile is 0. But when you compare it with our magnetic particle based system, it is coming, it is giving a quite decent amount of RIN profile. So this is actually a representative graph of RIN analysis to show the 18S and 28S bands. So this is actually a representative gel image of cDNA converted, RNA converted to cDNA and the PCR amplification of the cDNA. And the cDNA was amplified using Mature's K specific primer, and that is coming around 900 base pair of size. And this is to show that whatever RNA we are getting is downstream compatible. It is devoid of any kind of contaminants like any of the contaminants we have already discussed. That is to represent that is to show that it is downstream compatible. So to conclude that, we have almost screened 50 plus plant species, and the plant species includes different tissues as well, like as I've mentioned, like leaf, seeds, seedlings, or different tissues where they are included in the 50 plant species we have screened. And the total RNA yield that ranged from 3 to 110 microgram from 100 milligram of tissue. And the high quantity and intact RNA was recovered with minimal genomic DNA carryover. And you have seen that the purity profile were always greater than 2. That is always necessary for the ideal RNA extraction. Then the RIN value was coming above 6.5, and this is not the cutoff point. For some samples, some difficult samples alone, we were going till 6.5. For other samples, we were getting the RIN profile of 8, 9, or even greater than 9. Then the whatever RNA we got was downstream compatible, and our protocol is always, since we are using a magnetic particle based system, it is amenable for automation. So that is all about our work. So I would like to thank everyone in MagGenome for giving me the opportunity, and thank you, sir, and thank you.
Thank you, ma'am. We are happy to announce the launch of the Express RNA Plant Kit for plant RNA extraction. I request our Honorable Chief Guest, Padma Bhushan Dr. T. Ramaswamy and Professor Upadhyay to come up on the stage for the product launch. I'm honored to introduce our next speaker, Professor Aruna Goswami, Agriculture and Ecological Research Unit, ISI. His research interests include nanobiology and nanobiotechnology, more specifically malarial parasite culture, immunology, immunofluorescence microscopy, confocal microscopy, and genomics and proteomics tools. He was awarded with the Professor A.P. Odom Gold Medal by the International Society for Ecological Communications. He has 164 publications and 13 patents and serves as the editor for Nanobiotechnology, Basic and Applied Aspects. May I now request you to address the gathering, sir?
Hello. Hello.
Sir.
Hello.
We can hear you, sir. You're audible.
Yes, yes.
Entire screen.
This one.
Sir, can you please share your presentation?
You need to click on bench. Sure. Lunch.
Launch, sir. Launch. Sir, can you switch the tab to the presentation?
Yeah.
Yes, sir.
Can you see it, Kamalika?
Yes, sir. Can you go to the slideshow mode, sir?
Yes, slide.
Put it in the slideshow mode, we can all see it.
Yes, sir. Now it's fine.
Yeah.
Okay, so thank you, MacGenome, for organizing this 1 day or 2 day, I don't know, webinar. I have been working with nanoscience since 1997, and I have seen the development of nanoscience research in biology in India, being a task force member of DBT, DST, and ICAR, CSIR. So the title of my talk today is Nanome versus Genome. Are we Destiny's Children? When I was doing postdoc, the wind was to follow James Watson for his initiative in Human Genome Project. And in Human Genome Project, if you read the original document, what you will find that they have promised a lot. They had promised a lot, like they will bring gene therapy, they will give personalized medicine. Nothing has come out. So there is a backlash now among the US taxpayers that why we should fund this kind of project where no outcomes are coming. Okay. So now the sales are basically micrometer in range. So you cannot insert a micrometer size organelle like mitochondria or Golgi. Most of the particles, I will say, natural particles which are there, living particles are inside the cell, are of nano size, at least in one dimension. We do not call them classical nanoparticles, but we have classified them as a natural nanoparticle. So if collection, this is very important. If this is the dominant view that collection of genes in an organism is called genome, we can safely call all the nanoparticles present in a cell, natural nanoparticle. It's called Nano.
Hi, sir. Sorry to interrupt. Can I make sure that are you in the first slide?
Yeah, I'm in the first slide.
Okay, okay, fine. Go ahead.
So, so the nanome is nothing but it's a collection of all natural nanoparticles. So today, up to I worked for 20 years on synthetic nanoparticles, which are all published. So please read them. You will understand after listening this talk in a much deeper way those publications. For 20 years, I worked with synthetic nanoparticle, engineered synthetic nanoparticle, and we tried to see whether they can become a part of natural nanoparticles, but they couldn't act like that. So what I did sustainability analysis of this our work on synthetic nanoparticle. We actually repeated these experiments in my postdoc friend, Professor Volker Hessel, who is the Dean of University of Adelaide in Australia, and Professor Mark Excriber, who works on the University of Leeds in Spain. And three of us, we have published a paper this year showing that all the results which we got in India from our laboratory stands good in the laboratory of Professor Volker Hessel and Professor Mark Excriber. So that was a big satisfaction for me. Now, what we are now working for last few years is that we are using natural nanoparticle combinations coming from, or let's say nanome from medicinal plants to make nano gold and nano silver nanoparticles of various shapes and sizes. So if I go to how we make it, it's a very simple one. For I'm showing it for nano silver because the talk time is 15 minutes. So what we do, we take 1 millimolar silver nitrate and we take Withinia somnifera or Embolica officinalis or Camellia cebula. These are all medicinal plants, very common medicinal plants, in 2:1 ratio. And we constantly stir at 60 degrees centigrade for 30 minutes, and then there will be color change. And it will be you will see that if you do UV spectrophotometry, you will see that the peak is coming for nano silver. And then what we do, we take actually aqueous PEG 400 solution above pH 8, and we cap the nanoparticle so that it becomes more stable in biological system. And this we do by using formic acid solution. And I can, if you write me an email, I can send you the detailed methodology. And we then drive out all the leftover nanomes of these medicinal plant and clean it, and we basically stabilize it in water. And this is some example of nanoparticles stabilization with UV visible spectra. And then we do a large number of biophysical characterization of these nanome derived nano silver or nano gold using FEG tame. FEG tame is the best one. Field emission gun transmission electron microscope, field emission scanning electron microscope, UV visible spectrophotometry, XRD, DLS, EDAX with jitter potential, FTIR, and AFM, etc. And we show you some picture sample representative that this is the nanoparticle which we got. This is DLS data. So usually DLS, usually not. DLS actually measures the hydrodynamic radius. So the peak which you get at 120 or so in transmission electron micrograph, you will be getting half of the size. So we got very nice nanoparticle. Some of the pictures of these nanoparticle, let me show you. Silver nitrate nanoparticle from Withinia somnifera, which we call Orshogandha. Orshogandha in Ayurveda. Then this is the silver nanoparticle got from Horitoki, and this is for Amla. So you see in all three of them, different size and shapes of nanosilver. So we have now generated 400 different kinds of nanosilver and nano gold nanoparticle, and we are testing on a large number of infectious diseases. So let me show the data. So we are looking at mechanism of action of these drugs and efficacy on Bacillus coagulans, which is a beneficial probiotic bacteria. We look at Vigna, its effectivity in Vigna radiata, which is a plant. So this is the phytotoxicity for studies needed for patenting these drugs. And all the data which I am presenting today are unpublished. They are not published. So we use a detrimental bacteria called E. coli and two fungi which makes human skin disease. Trichophyton, two species that we take, and we have tried and successfully got results from in vitro model of diabetes. So this is actually the effect of these nanosilvers on Bacillus coagulans and E. coli, and you will see that they are very effective on these organisms. This is actually effect of nanosilver from Amla, Embeddica officinalis, on Bacillus coagulans and Escherichia coli. And let me tell you a secret. So all the people who work for homeopathy and allopathy, Ayurveda, they actually use plants, medicinal plants from different areas, which will have the batch to batch variation, and then they actually try to produce nanoparticles from them. We didn't do it. We did a very nice shortcut. Actually, we took homeopathic mother tincture. Homeopathic mother tinctures are nothing but ethanolic extracts of medicinal plants. So ethanolic nanome of those medicinal plant, and we took Ayurvedic churna, Orshogandha churna, commercial product, commercially, with nothing. We did it by collecting that plant from wild or somewhere in Delhi, Not that. We took the commercial formulation and we showed that they can act as a good framework material. So this is the IC50 value of Holitoki Churna, but we are calling it as aqueous extract of Holitoki or aqueous nanome of Holitoki. It's a new subject, so I'm just taking little time. So you see in the diabetes model, as acarbose is the standard drug, and alpha glucosidase is the does give this much of response, and this shows that our product works very well in comparison to acarbose. So basically, we are developing allopathic nanomedicine. This probably you have understood that we are making allopathic medicine, basically new allopathic medicines based on nano gold and nano silver. And then you can see the range. The number of homeopathic mother tincture available everywhere in the world, it will be more than 400 to 500. So at least 400 to 500 different kinds of nano gold and silver we will generate. This is the bigger
Sorry for
picture.
the interruption, sir. Since we are running out of time, can you conclude it in a couple of minutes, sir?
Yes. So this is IC50 value from ashwagandha, and the conclusion is in this present study we have formed nano gold and nano silver based allopathic medicine, and these are some of our publications. And this is a little bit of work which we are presenting for the biosafety assay we have done. We have done all kinds of phytotoxicity and other assays on the model organisms. From Arnica, we got the same following similar protocol, but these are process patented now. And we characterized also them. And these are the nanoparticles which we got from Amla. And you see nice ball on the left hand side. And these are basically phytotoxicity assay and on different parts of the plants. I will not go into detail. So this is from Atropa belladonna. So this also we make it, and these are the characterization. And this is Trichus, Nux vomica, and this is the dose dependent variation, and this is the end of my talk. So one concluding remark I want to make is that during late 1990s to up to 2014, various chambers of commerce in India, FICCI, PhD Chamber of Commerce, they actually worked for nanoscience industry, and we have given enough number of lectures to them, a lot of lectures. So I will request MacGenome to raise the voice that what about talking about nanome research? And they can contact MacGenome, can contact FICCI, ASSOCHAM, all these industrialist lobbies who can talk to the Prime Minister or and generate funds, because most of the companies, including MacGenome, they are good companies, but they are not getting any business. We are not getting from 2014 good amount of money so that these organizations like MacGenome, they can proliferate. And I wish with this serious concluding remark that Kamalika will take a center stage for asking lobbies who can influence government to give money for nanome research. Thank you, and have a good day.
Thank you for those thought-provoking words, sir. It gives me immense pleasure to invite our next speaker, Dr. Oliver Strobuck, Laboratory Head, Laboratory of Metabolomics, Comenius University in Bratislava, Slovakia, who focuses on the biochemistry of iron oxides mineralization and accumulation during pathological processes in humans, including neuroinflammation and neurodegeneration. His research activities include biogenic iron oxide nanoparticles from the origin of life point of view, over diagnostic possibilities using MRI techniques, up to therapeutic applications in human biomedical research. His skills and expertise include MATLAB, diagnostic nanomedicine, MRI, bioelectromagnetics, medical physics, molecular biophysics, among others, with more than 50 research publications. I request you to address the endeavoring young mind, sir. Can you hear us, sir? I think sir will be joining in a moment. Can you hear us, sir?
I can hear you, and you?
Yes, sir. You're audible,
Oh,
sir.
finally. Okay.
We request you to share your presentation, sir.
You can see my presentation?
Not yet, sir.
Not yet.
There will be one slight tilted arrow in the down. So click that and go for screen share and open your presentation. Hi, Dr. Oliver. So there will be one slight tilted arrow in your screen, down.
Hello, hello.
Ah,
And now you can see my
yes,
presentation.
now we can see.
Okay, and you can hear me. It's perfect now. Okay. So, thank you
Yes.
very much. Thank you very much for your very kind introduction, and good afternoon to India, although it is still morning here in Slovakia. First of all, I would like to thank for the opportunity to introduce our research at this excellent and I believe that also fruitful event. So thank you, it's my honor. In my talk, I will present our research and results related to the determination of MRI relaxation properties of magnetic nanoparticles intended for biomedical applications. Here is the outline of my talk. I'll start with the question, who are we? Next, move to our motivation to study magnetic nanoparticles. I just briefly touch MRI relaxometry terminology, and finally our results related to the synthetic magnetic nanoparticles and ferritin derivatives. So, who are we? Let me start very briefly by introducing our young and small laboratory. Laboratory of Metabolomics is part of Biomedical Center in Martin, which was opened in 2016 as a research base of Jesenius Faculty of Medicine in Martin, and laboratory officially exists only from 2020. Laboratory infrastructure includes mass and NMR spectrometers and MR scanner for preclinical imaging, spectroscopy, and spectroscopic imaging. And our research focuses on targeted and non-targeted metabolomic analysis of various diseases, including COVID-19, cancer, and neurodegeneration. Also in preclinical studies of ferritin derivatives and theranostic applications of magnetic nanoparticles, and in close cooperation with University Hospital in Martin, also in clinical studies of multiple sclerosis and human brain iron imaging. In research related to the magnetic nanoparticles, we focus mainly on MRI relaxometry of synthetically prepared and biologically formed iron oxide nanoparticles, including ferrihydride, hematite, and magnetite. Partly, we also deal with magnetic hypothermia, and we are also interested in the question of in vivo hazard related to the use and existence of magnetic nanoparticles within the human tissue. So what is our motivation to study magnetic nanoparticles? It's strongly associated with the wide range of possible biomedical applications of magnetic nanoparticles, and biocompatible magnetic nanoparticles are currently a highly desired tool or material for various biomedical applications, including as drug delivery systems, which involves chemotherapy, gene therapy, and photothermotherapy. Also for separation of biological molecules, which includes also enzyme immobilization, in magnetic hypothermia, and also in magnetic resonance and magnetic particle imaging. Magnetic resonance imaging is the best biomedical imaging technique currently available, I guess. I'm not going into details of MRI principles. I just mentioned 4 basic MRI parameters that modulate MRI, that modulate the weighting of MRI images. And first parameter is repetition time. It's time TR. It's time between successive pulse sequences. Next is echo time. It's time between the delivery of radio frequency pulse and the receipt of the echo signal. T1 is longitudinal relaxation time related to spin lattice relaxation, and T2 is transverse relaxation time related to spin-spin relaxation. And by modification of repetition time and echo time, we are able to receive different weighting of MR images. For example, for short TR and short TE, we obtain T1 weighted images, and for long TR and long TE, we obtain T2 weighted images. In MRI relaxometry, we generally use the following pulse sequences, like T1 mapping rare protocol, T2 mapping multi-slice multi-echo protocol, and T2 star mapping multi-gradient protocol. Relaxometry is associated with relaxivity parameter, and relaxivity defines MRI contrast efficiency of magnetic nanoparticles and is calculated from the slope of inversion relaxation time dependence on iron concentration. Okay, let's move to our results. Right, just briefly touch our recent results related to MRI relaxometry of synthetic and biogenic magnetic nanoparticles. We tested several magnetic nanoparticles with different coating, different ligands on their surface, and all tested magnetic nanoparticles were prepared at the Institute of Experimental Physics, Slovak Academy of Sciences in Košice. And we were especially interested in the relaxivity parameters of these specially treated magnetic nanoparticles, and on the ratio of transverse and longitudinal relaxivity, which defines the prevailing relaxation mechanism of magnetic nanoparticles. So whether particles will produce hypointensive which mean negative contrast, or hyperintensive, which mean positive contrast. I will go very briefly through the results and I will compare them at the end. Um, Here are example of chitosan stabilized magnetic nanoparticles and the corresponding parameters. Example of polylysin stabilized magnetic nanoparticles with corresponding relaxivity values and relaxation and ratio of relaxivities. We calculated also relative contrast for each magnetic nanoparticles. Magnetic nanoparticles stabilized by different amino acids, including lysine, glycine, and tryptophan. Here are parameters of relaxivity ratios. Here's the example of relaxation time maps, longitudinal T1 map and transverse T2 map with increasing gradient of iron oxide nanoparticles. And finally, dextran stabilized magnetic nanoparticles with corresponding values and relaxivity ratios. So let's compare our results of relaxivities and ratios in comparison with commercially available magnetic nanoparticles, which are also based on iron oxide to produce hypointensive signal. We are interested mainly in transverse relaxivity, and as you can see, especially for chitosan and polylysin magnetic nanoparticles, our values are much higher compared to commercially available magnetic nanoparticles, which means that our magnetic nanoparticles produce better contrast for MRI, which motivates us to next studies on this issue. Okay, let's move to biologically produced magnetic nanoparticles, and probably most interesting, most studied, and well known is ferritin. So just briefly, what is ferritin? Ferritin is in protein envelope with central cavity and mineral core inside in the form of crystalline ferrihydride. And the main function of ferritin is the elimination of toxic ferrous ions when they reach critical concentration for the cell and depositing them in the form of non-toxic ferric ions for the later usage by the cell. And what is crucial for our applications that, at least in humans, mineral core composition depends on health condition. And indeed, in 2004, Quintana and colleagues found that ferritin from patients with Alzheimer's disease is structurally different from physiological or native ferritin, starting to refer as a pathological ferritin. And they found that physiological ferritin consists mainly of ferrihydride, less amount of hematite, a very small phase of magnetite. And on the other hand, pathological ferritin consists mainly of magnetite, less amount of wustite, and very small phase of ferrihydride. And this conclusion were confirmed in 2017 by using muon spin rotation technique. In addition, ferritin has been proposed as a precursor of iron accumulation in human tissue, and elevated levels of iron in humans are strongly associated with various pathological processes like neuroinflammation, neurodegeneration, and even cancer. But especially with neurodegeneration, iron accumulation is connected to magnetite, nano-sized magnetite mineralization in the tissue. And here you can see the very first picture of human brain magnetite from the Kirsching publication from 1992. So on one side we have physiological ferritin with ferrihydride like mineral core. On the other side, we have pathological ferritin with magnetite-like mineral core, probably because of disrupted iron homeostasis. What is crucial for us that magnetic moments of these two structures differ, and magnetite from pathological ferritin cause so-called hypointensive artifacts in MRI, which enable to distinguish these two structures and to study MRI relaxivity of magnetite from pathological ferritin and biochemistry behind the transformation of ferrihydride-like core to magnetite-like core. We use so-called magnetoferritin as a model system of pathological ferritin, and magnetoferritin consists of apoferritin and artificially added phase of magnetite. It's nano-sized crystals. And in this research, where we use magnetoferritin as a model system of pathological ferritin, we have two main goals. First is diagnostic. We would like to develop non-invasive MRI diagnostic methodology of pathological processes that are associated with biogenic iron accumulation and mineralization. Because, for example, in neurodegeneration, iron accumulation and magnetite mineralization probably precedes the clinical symptoms of the disease. And second goal is therapeutic goal. We believe that understanding of the biochemical processes that result in biogenic iron accumulation and mineralization can lead to the understanding of pathology. Here are some preliminary results related to these two goals, but let's move to the results of MRI relaxivity of native and magnetoferritin at 7 Tesla. And as you can see, for all relaxivity parameters, we are able to clearly distinguish native ferritin from magnetoferritins with different iron loading factors. Same situation is also for the ratios of relaxivities. One anomaly is here for your 2-star ratio, where we can see the increase in the value for native ferritin, and we still don't know the reason, and we hope that in the near future we'll be able to understand them on the molecular level. Okay, conclusion. So all tested magnetic nanoparticles which were prepared at the Institute of Experimental Physics with different coating, with different ligands, show excellent hypointensive MRI contrast properties compared to commercially available magnetic nanoparticles based on iron oxide. So they have prevailing transversal relaxation, and it is possible to clearly distinguish between native ferritin and magnetoferritin by simple MRI relaxometry, which can be helpful in non-invasive diagnostic of pathological processes that are associated with iron accumulation and magnetite mineralization in tissue. Okay, thank you very much for your attention. I would like to thank my colleagues from Biomedical Center and from Institute of Experimental Physics. I'm open to answer all your questions, and we are very open to any kind of cooperation. More information about our center you can find at this link, and here's my email, so do not hesitate to contact me. Thank you very much.
Thank you for sharing your knowledge with us, sir. Questions can be posted in the chat box by the attendees. They'll be answered. Thank you, sir. I
Thank
request
you.
everyone to break for lunch and get back by 2:20 to continue the session. I repeat, I request everyone to go for lunch and get back by 2:20 sharp to continue the session.
Go out, take left corner to the end.
So lunch is being provided in the left corner, just if we take left after getting out of this room. Dead end of the left. I'm delighted to invite our next speaker, Dr. Shejal Shah, Head, Department of Computer Sciences and Biosciences, Marwadi University, Rajkot, India. He's an associate member of American Association of Cancer Research, life member of European Association of Cancer Research, scientific recorder at COVID-19 Omics Research Consortium and Breast Cancer Research Consortia. She has been selected in top 10 women scientists and awarded Indo-US Women in STEM Fellowship 2019. She has 6 Indian patents, more than 25 publications, 8 book chapters, and 8 research products to her credit from various funding agencies. I request you to address the audience, ma'am.
A very good afternoon, everyone. Don't worry, I won't let you sleep. It's a general talk. So, first of all, I would like to thank the team MacGenome, especially Ramchand sir, for providing such a wonderful opportunity. So, let's go for the recent trends, or we can say current trends of nanotechnology. Can we quote one line for nanotechnology that smarter solutions for the bigger problems? Yes. So if we go down the line 20 years back, between 1960 and 1980, the nanotechnology, it was flourished and it has surged like anything since last 2 decades. Okay, if we go to the 2000, passive nanostructures, see, some nanoparticle synthesis and coating has been developed, followed by active nanostructures were developed, integrated nanosystems, then molecular nanosystems, and certain technologies were came into existence. There are different types of nanotechnology. We can say dry nanotechnology and wet nanotechnology. For wet nanotechnology, the biological samples when we used, if we are synthesizing some nanoparticles from microorganisms, from some bacteria or some from fungus, which include the humidity, we can say it's a wet nanotechnology. And when we are dealing with the silicon or carbon, it's a dry nanotechnology. Certain approaches are there, top-down approach and bottom-up approach is there. Nanotechnology has a divergent applications. If we say different types of application in different field, if we start with the energy, as we know that batteries that can last longer than medicine and drugs, especially if we talk about the cancer medicines, drug delivery system, then nanobiotechnology, certain medical devices are there, optical engineering, defense security, organ engineering, tissue engineering, then special zinc oxide nanoparticles, titanium oxide nanoparticles, they are high in demand with the cosmetic industries. Then smart fabrics are there, dust-free and stain-free fabrics, which is having the antimicrobial and antiviral properties, especially the gene wares. Okay, so these are the applications. If we talk about the spatially, I'm the cancer person. I'm not the nanotechnology person. Just before 6 years, one student came to my lab. She did her MTech from SRM and she told me that, ma'am, I'll do PhD under your guidance, but that too in nanotechnology. So I learned many things from her. I am working on cancer transcriptomics, proteomics, metabolomics, everything. But yes, we can say there is a strong role of nanotechnology in cancer diagnostic as well as prognosis. Okay, diagnostic kits, we are working on one diagnostic kit for the oral cancer. In the prognostic, some prognostic biomarkers we can link up with the nanotechnology. Especially the scaffolds for the organ engineering and tissue engineering for the bond treatment. We can go for this drug delivery system. As we know, all these things, it is high in demand. CNF, carbon nanotubes. They are mainly useful for the gene delivery, gene editing, organ tissue engineering, and organ engineering. Carbon nanotubes, especially if we say for biosensor and diagnosis purpose. Trojan horse nanoparticle that can eat debris related to the plaques. If you have some blockage, this nanoparticle simply it will eat the plaques. Nanotech detectors for heart attack. This is the biosensor machine that can detect heart attacks before 10 minutes. Yeah, it's fine. So main thing is, apart from, see, I'm highlighting the medical part because I'm working on that part. So there is a strong role of nanotechnology in medical devices and in biomedical side, right? But apart from medical side, there is a strong role of nanotechnology in agriculture, in environmental sector, in textile industries, in food industries. Say, for example, in textile industries, my lab, it is working on antimicrobial and antiviral cotton fabric, and we have started making masks, antiviral masks, especially anti-COVID masks. So it has started in our lab. Then we are dealing with the strawberry coating as well. We have synthesized some zinc oxide nanoparticles which have a strong efficacy that can last up to a strawberry post harvesting technology. We can say if it can go with the post harvest technology. Okay, nanosponge. Nanosponge, these are the polymers that can remove the toxins from the bloodstream and it will purify the blood. Nanoflares, that are the single stranded molecules that can go within the cancer cells and it can target the cancer cell with the high expression of specific genes. Okay, so you can identify, even you can deliver the stem cells as well with the nanoflare. We all know about the nanorobots, okay, or we can say nanobots. So it has a crucial role in biomedicine, spatially, and nanorobots. It will be mainly useful for to remove the kidney stones. Urologists, they are nowadays using this nanorobots for gene editing as well. And this nanobots mostly for endoscopy, they are using, clinicians, they are using for the endoscopy. Even a nanobot mouth wash has been envisioned, which would be nanobots clean up food particles if you have stuck into your mouth. It can easily, it can be used as a mouthwash. Yes, smart pills. For imaging purpose, for biosensing purpose, we can use these smart pills for drug delivery purpose as well. Yes, we can see the market size of nanotechnology here. It may exceed $125,000 million in the next 5 years. And you can see the growth by 2030, it will be 964 US billion dollar. So apart from, as I said, from medical background, apart from medicine, it has a role in divergent sector. If we talk about the environmental sector, dye degradation, water degradation, oil degradation, plastic degradation, everywhere there is a role of nanotechnology. Textile industries, the dust-free and stain-free gymwear scientists have developed, and they are in high demand. For food, nanobiosensors that can easily detect the pathogens from the food. So it is already developed. In agriculture, nano biofertilizers are there. Then edible biocoating, as I said, it's in the market already. Sensors for foodborne pathogens, emulsions. It can be used titanium dioxide. Mostly scientists are using. So as in the morning, sir said that nano mission did a great role, played a great role in terms of they have given us the fund. I am also the awardee of DST fund, and using those funds, we have filed certain patents, and we can say that the mark in 2007, NSTI launched a 5-year program that is Nano Mission, and it was about 1,000 crores. So basically, you can again you can apply and you can build your infrastructure. Certain challenges are there. If we talk about the nanotechnology, obviously the safety and environmental hazards are there, ethical issues are there. So we need to deal with such type of problems. Publications, our collaborators, where we are working for the nanotechnology and cancer genomics. Thank you so much.
I would like to welcome our next speaker, Professor Deepthi Menon, Professor, Amrita Institute of Medical Sciences and Research Center, Kochi, India, who is a researcher working in the field of nanomedicine and nanodrug delivery. She has more than 15 years of experience in the field of nanomaterials development, engineering, and applications in diagnostics and therapy. Her research interests include development of nanomedicines for targeted cancer diagnosis and therapy, tissue engineering, and drug delivery using nanomaterials. She has more than 100 peer-reviewed publications with 8,114 citations.